Okay, so I think we can get started. Thanks everybody for coming today to this event. Our first speaker is going to be Paul Suska, who is the supervisor of the Planning Protection and Assistance Program here at DES. Paul?
Good morning, everyone. So this, in case you're wondering if you're in the right place, this is the CyanoHAB workshop and tabletop exercise being presented by the Department of Environmental Services and U.S. Environmental Protection Agency. Thanks, Liz, for putting this all together. So to give you an overview of what will be on the agenda, you all have a copy of the agenda. I'll just kind of be hitting the high points here. I'm going to be talking for a little while briefly about our Cyano Bloom response protocol that we developed at DES, and then there'll be a presentation on the basics of what you need to know about cyanobacteria and monitoring. Since there's so much information, the focus will really be on what are the essentials, and then information about some tools that were developed by US EPA for water systems to develop their own customized plan, it's also a recommendation that we have. We think all water systems really should develop their own plan. And then some information about the toxins that are sometimes produced by some cyanobacteria under certain circumstances. A case study from Salem, Oregon will be instructive about the way things could head and also help us as we go through the tabletop exercise. Then there'll be a 15-minute late morning break, and we'll come back for another presentation on treatment at public water systems for cyanobacteria and cyanotoxin removal. And then what you've all been waiting for, the tabletop exercises, which will take 2 hours total, but there's a 30-minute lunch break in there. So 90 minutes for the exercises and 30 minutes for the lunch break. And then we'll come back and review the exercise, how it went and what was learned and so forth. So just quickly to review, why should water systems be concerned about cyanobacteria and cyanotoxins? Well, there's immediate concerns and there's long-term. I mean, obviously, sometimes they produce toxins, and those toxins can be a health concern, but even if they're not at harmful levels, it can be harmful to consumer confidence, which is something that water systems work so hard to maintain. And then, a lot of cyanobacteria, even if they're not producing toxins, do produce taste and odor compounds. And of course, that can mean big headaches for water systems. And cyanobacteria, particularly when there's sudden changes in population, when there are blooms, it can affect your treatment in a number of ways. pH changes, increased demand for chlorine, increased turbidity, shortened filter run times, and also increased formation of disinfection byproducts. Then there's the longer term concerns. These blooms, if they're occurring repeatedly, can drive the transition of your lake or reservoir from being a low nutrient, relatively clear, low turbidity water body to one that has higher nutrient levels, more turbid, more issues with disinfection byproducts as well as turbidity. So for those reasons, you want to take some measures to actually prevent blooms. There's also a possibility that some of the toxins will be regulated in the future, although they aren't right now. So what is the regulatory status of the cyanotoxins? As I said, so there's no maximum contaminant levels that have been established either in New Hampshire or on the federal level. Although EPA has issued health advisory levels for exposures up to 10 days. And they've issued these health advisory levels for microcystins as a group. There are a number of different microcystins, and those are toxins. But the levels they've established are microcystins as a group, and then also another set of advisory levels for cylindrospermops and different levels for 6+ and under 6 years old. Certain cyanotoxins also made the contaminants candidate list, the CCL4, and in addition to cylindrospermopsin and microcystin, also anatoxin A and saxitoxin. So that means that the EPA is considering establishing, regulating them in the future. And then, as you probably know, there were 10 cyanotoxins that were included in the contaminants that had to be monitored under the unregulated contaminants monitoring rule that went from 18 to 2020. So stay tuned for the regulatory status. This bar chart shows the number of confirmed cyanobacteria blooms in public water supply sources in New Hampshire going back to 2010. And you can see the numbers are not really big, and nor are they very consistent. And if I asked Excel to draw a trend line, and it drew this line here, which is not a very convincing case for any kind of trend in New Hampshire. So really, they've been sporadic. And the thing is that our numbers are subject to the fact that awareness has varied over time. There may be any number of blooms that have occurred that we just haven't become aware of. So again, stay tuned. All right, so we developed this CyanoHAB response protocol for New Hampshire, and we did this in response to really a national trend toward more severe cyano blooms that have affected some water systems elsewhere in the country, but as close by as Lake Champlain. Again, it's not clear at all that there's any kind of trend in New Hampshire, but it's an evolving situation. I think that it's, my own view is it's only a matter of time before this becomes an issue for at least some of the sources in New Hampshire. But what's more important is our understanding is evolving in terms of cyanobacteria, what conditions lead to blooms, under what conditions did they produce toxins or even harmful levels of toxins and so forth. So we tried to provide some very workable guidance for our water systems to follow amidst all this evolving situation. And it's also challenging because there's so many different types of cyanobacteria, so many different toxins, and so many different, it's sort of source specific in terms of knowing when and whether there might be a bloom in your source. So we felt that there was a need for very practical guidance for water systems in New Hampshire, and both to be prepared and even if you haven't developed your own response protocol, this is something that you can follow so that you can respond if there is a crisis, if there is a bloom. And because not only might there be a health concern if toxins are detected at harmful levels, but some of your consumers are going to have health concerns if there's a bloom or even an appearance of a bloom. So really, in order to help you maintain consumer confidence, it's good for you to have this guidance, we think. The sort of key, the essential part of the guidance document is this flow chart. And we've worked to keep it simple. And you can see that there's some color coding there. The white boxes, these are the steps that the water system would take. And then we have steps here that would be taken at DES. And then finally, what would have to go out to another lab. So focusing in first here on what are we asking water systems to do? The main thing is that we're recommending all water systems that have surface sources to take a good look at your source at least every day. Look for signs of algae blooms or cyanobacteria blooms. Get to know what cyanobacteria blooms might look like. And particularly if the source has a history of suspected cyano blooms, we strongly recommend that you do more than simply looking at your source every day. And that's something that Amanda will talk about later on. So if you see visible signs of a bloom or scum, or if there are water quality parameters that you notice in your plant that indicate a possible bloom, then that sort of triggers, okay, what's the next step? And the next step is for you to sample. We suggest you use HDPE brown bottles, which Amanda's lab can supply. We recommend that you sample the bloom or the scum itself, some location in the open water that seems to be outside the bloom. Also sample the raw water and also the finished water. We aren't necessarily going to be analyzing the finished water or even the raw water for toxins, but it's good for us to have those samples on hand from the very beginning in case what we see in the bloom and in the open water indicate that there's that it makes sense to look for toxins there. So again, the next steps become DES's responsibility. We suggest that when you take those samples, you get in touch with the Jody Connor Limnology Center, Amanda McQuaid at the Jody Connor Limnology Center at DES. Amanda's lab will put those samples under a microscope, identify whether it's cyanobacteria, if so, what kind, and also get an idea of the density of the cyanobacteria. And that's a really important parameter in terms of decision making. Amanda and her lab will consult with the Drinking Water and Groundwater Bureau, and together we'll figure out whether it makes sense to do toxin analyses or to recommend that you continue sampling or whatever the next steps ought to be. And as I said, there's some work that can be done in the Jody Connor lab, which includes not just what I described, the identification and the enumeration, but also they can do ELISA tests for total microsystems. There's other tests that can be done for specific cyanotoxins, but we don't have the capability at our labs to do that, and that would have to go outside. And based on what Amanda's lab is seeing and what results would come back from another lab, we would circle back, consult with you, and make recommendations about what next steps ought to be, whether it's simply more sampling, adjusting your treatment, or public notification, depending on the severity. So that's basically the protocol, and I'll be handing it back to Liz right now.
Thank you, Paul. Next up, first speaker, we have Amanda McQuaid, and I will give her the controls while I introduce her. Good morning, Amanda. So Amanda
Good
McQuaid.
morning. Can you hear me?
Yes, and we can see your presentation.
Great,
So
thank you.
Amanda McQuaid is an environmental scientist in the biology section for the Watershed Management Bureau. She oversees the newly formed harmful algal and cyanobacterial bloom program, as well as the beach inspection program for New Hampshire DES. Amanda researched lake ecology and toxicology at the University of New Hampshire and is dedicated to outreach and education on the subject of harmful cyanobacteria blooms. And Amanda, whenever you're ready, thank you.
Great, thank you very much. Okay, so yes, my name is Amanda and I'm with New Hampshire DES, and this phone number here is our cyanobacteria hotline, and we also have a email specific for our HAB programs. And this is just a primary way that we receive our complaints in New Hampshire. So, the highest number of recreational advisories in New Hampshire have been issued in recent years, just, right over the 30 to 35 threshold. Last year in 2020, we did have some sort of setbacks with the pandemic and our programs, and we didn't have as much staff and help. However, we sort of changed our messaging to also put out alerts. So in the event that there would be a bloom, someone sent us a photo, we could clearly see that there was a bloom at the beach. We might drive all the way out there and it's already dissipated from the location and otherwise appears clear. So we have these advisories when we know that there's an ongoing bloom that are exceeding the 70,000 cells per milliliter threshold, but we will also share information locally with people that live on the lake, associations, or the town or beach manager, just to let them know that they should be on the lookout. Advisories and alerts are the same in that way. We will never close a beach or a water body totally. We just are trying to inform the public of the severity of the bloom. With advisories also, once we know that there's an advisory going, we will follow up weekly at the latest. It depends on what other blooms are going on, how busy we are, and who can help, but we will keep up with our weekly sampling to just check on the status. In 2021, we are still in the season. You wouldn't think it by looking outside, but there are still blooms and we still have current advisories. So the 2021, final results are really not ready yet. Looking back, beyond, the last 10 years even, we did have another year. 2008 had an increase in reports of blooms. Really can't say exactly why. It could be a combination of things, of increased public awareness, certain meetings and campaigns that year that might have helped. But overall, there are wet and dry years that can create these sort of patterns of bloom surfacing to our, at least us seeing it. Sometimes we assume that the water looks clear, it's all good, but there's also blooms that can be, down in the metalimnion or even deeper in some cases that will pop up at certain times of the day. And so these blooms are sort of sporadic events in a lot of ways. And this graph shows sort of the number of advisory days over time, but this is not a significant trend. This is just, maybe if we looked back just to 2010, there would be a significant trend, but we know that there's sort of these years where we have more blooms spotted, and those reasons could also be due to public awareness. And the blooms we see in New Hampshire are not that severe, but they don't always just look like a blue-green cloud in the water. Most concerning is when the water itself just has a green hue and there's not a surface scum. If your water is overall clear and then all of a sudden looks green, you are likely seeing the start or the height of a cyanobacteria bloom. Once they surface, they get caught in the surface tension sometimes, or along a shore or a beach. And that's when we see them piling up. So they're growing in the lake, but they're coming up and they're getting piled up in a cove, a beach, someone's shoreline. But there's a lot of different species, and there's a lot of different types and habitats, and they all have sort of different requirements for growth. So be on the lookout, but note that they're not always going to just be these blue-green scums that we typically see. And sometimes cyanobacteria are mixed with another organism that can kind of change their colors a little bit, that makes the bloom look sort of these odd orange clusters. Like this top right corner is actually like an amorphous blob that was formed with Warrenichinia and Botrychococcus. That microscopic picture up there with the orange arrow created like this sort of blob, and when you touched it, it dissipated. It also created sort of this yellowish orange streak on the surface of waters. So, aside from the toxicity of it, it can be alarming for people just recreating in the water to see these things. We also have benthic mats that can come up at times and then shore beyond the beaches, and they could be a danger, probably more so for people that touch it, maybe get a skin irritation, but more so for dogs and pets that might be curious about what this stuff is. We also get reports of someone has poured detergent in the water. Sometimes blooms just start to look like white clouds when they're dying out. They're getting kind of bleached by the sun, they're losing their pigments. They can be really toxic at this time as well. They're releasing free toxins into the water. So at the moment, it could actually be, also a dangerous situation, even though the bloom is dying. Like I said, we also have these deeper, not benthic, but sort of blooms that are growing really deep in the lake that we just don't even know are there. And they'll come up at different times, and we'll get reports that there's paint chips in the water. And this is usually due to Planktothrix, a certain species that can regulate its buoyancy like many of the cyanobacteria do. They like to live deeper in the water at most times. There's also these like blue-green marbles called nostoc that are pretty rare, I would say. We don't really see them, but they're not going to create like a surface bloom, and they present sort of a different issue. There's also pico-cyanobacteria that are so tiny you really can't see them under the microscope. There's not a lot to report on the toxicity of pico-cyanobacteria, but there's a lot we don't know about them. So you need like an epifluorescent microscope that would shine. That's that red dot photo at the bottom. Once we can shine a light on them, they give their pigments back and we can actually count them and enumerate them. This is not something that we can do at DES, but there are some labs equipped with epifluorescence. But overall, we're looking at just such a huge variety of different types that could pop up in our lakes. And gliotrichia is one of those, and gliotrichia is another type that doesn't really look like a blue-green scum in the water, although it can get caught up in the surface accumulation and look like a scum. It's usually these little tiny dots, like little orbs in the water that people usually think are eggs. But gliotrichia is a very large cyanobacteria colony known to produce at least microcystin in low levels. So its size is something, but it doesn't, it's a low, as far as we know, it produces low levels of toxins. So even though it's very large compared to its size, it has sort of a lower level of microcystin. And these have been present in some of our water sources in New Hampshire. So if anyone sees anything anomalous, they think they're seeing cyanobacteria, we are encouraging them to contact New Hampshire DES. And so we receive a lot of reports and complaints over the summer. That number has, doubled in recent years. There's just a lot of people, I think, again, public awareness, realizing that they are not looking at pollen, that they're looking at a cyanobacteria bloom. People will give us a call at the hotline. We arrange sample collections. We do inspections just to see how extensive this bloom might be. We are also limited to the shoreline most times, so sometimes we get help from a lake association or a town representative to help us get out around the lake. Samples are then analyzed within 24 hours for cyanobacteria identification, and we also do a cell count. We use the 70,000 cells per milliliter threshold to issue recreational advisories. So these samples, if we confirm that this is an advisory, we would often subsample and freeze these so that we can later do our cyanotoxin analysis. And we do have an ELISA methodology that we could do at the DES. We've been really focused on microcystins. We're starting to get into anatoxins and cylindrospermopsin. We've also trialed BMAA and saxitoxin. So ELISA kits are available for all of these toxins that we are focused on. Again, the advisories are issued when there are confirmation that a bloom is extensive, that the cell count exceeds 70,000, and that it doesn't appear to be dissipating anytime soon. Alerts, again, are shared when maybe a bloom was spotted, but it dissipated very quickly and we can't find it anywhere. We would still go around to sample, but if it looked like it was just a small accumulation that maybe dissipated, we'll leave it at that and at least have the photos shared locally so people know what to look out for. And that's kind of what happened at Lock Lake this summer in Barnstead. We have different lake groups. We have a volunteer lakes assessment program. We have river trend groups. We have lake trophic survey groups. We have a lot of staff out on the waterways all the time, all summer. However, Lock Lake is no longer in the VLAP program, and it's no longer in the beach inspection program, but somebody at the lake noticed that the beach wasn't looking right. So we went out during the first week of August. Staff went out to collect samples, and nearly all the beaches had a scum that looked like this. The organisms were Microcystis, Warnachinia, and Dolichospermum. and over 50 million cells per milliliter were observed in the worst areas like this. Where it appeared clear, relatively clear, there were still elevated cell concentrations, but the beaches were being impacted by this bloom. Sorry, this slide is slow. Okay, so one interesting thing that happened with this system was that, well, surprise, Webster Stream was going to be pumped for the community for drinking water in the fall. And so this event being in August into September was kind of close to that timeline of wanting to go online for drinking the water. Webster Stream is the outflow of Lock Lake. Lock Lake actually used to be Webster Stream and was dammed up to be a lake. And it flows over a dam down to Webster Stream where the intake pipe is. And the public water system here used a multi-parameter sonde and put that at the dam to measure a few different parameters, but mostly focusing on the pigments associated with cyanobacteria. And in this case, we're focusing on phycocyanin and chlorophyll, which is a sensor that you can put on these devices. So they had this kind of hanging over near the dam where the outflow was to the Webster Stream. And you can see over time that there are just sporadic times where the phycocyanin levels increase pretty heavily. This is a unit, a relative fluorescent unit, which can also be calibrated to micrograms per liter. I'm not sure, this graph was given to me by the public water system, and I'm not sure which unit they are referencing here, but they're pretty much the same usually. So that's something to check with them. But what we can see here is that phycocyanin indicates cyanobacteria was at that location in high levels and at different times of the day, at different days during the course of that bloom event. And so this just kind of shows us in, not in real time, but looking back at, every, I think 10 or 15 minutes, what signal was being presented at that location. And there were days where maybe there wasn't a lot going over the dam, and other days where these heavy, thick accumulations were probably going right over the spillway. And so what's nice about Lock Lake is that it was part of the Volunteer Lakes Assessment Program. I think this is a report from 2019, and I do think that that was the last year that they were in the program. I'd have to check again with the VLAP program on what the status is, but maybe due to the pandemic or other things, but I don't believe that this lake is going to be in this program. However, it does have a lot of data already, and we know that it is a lake that is considered mesotrophic. Mesotrophic lakes are of moderate parameters of moderate phosphorus, chlorophyll, water clarity. So they're sort of that mid-grade type of lake that can be very vulnerable to blooms and eutrophication. The lake also has a pretty good flushing rate, which is good. However, it's being flushed to the Webster Stream, which is something we want to be careful and look very carefully at. But overall, this lake has been impaired for certain parameters that we need to really focus on, like phosphorus. So we know that there's a phosphorus issue in this lake that could be feeding these types of blooms. This lake is also impaired for cyanobacteria, but that was due to events that happened in 2009 and 10. And these impairments usually last for around 10 years until we realize that the situation has gotten better, or we don't have any data to support it, so it can be stuck in this category. Hopefully that will not be the case for Lock Lake. However, after the bloom event this summer, I don't think this will be removed from the impair list anytime soon. But these data are available on the New Hampshire DES website. It's especially useful if you go to our lake mapper to see water quality parameters for every water body we have in New Hampshire. So moving on to Arlington Mill Reservoir in Salem, this is an advisory that we're actually still working on. I sampled yesterday. It looked like conditions have been much improved, so we'll still follow up with them in the next week. But This was sort of the same situation as Lock Lake just about a month later. Microcystis warnachini and dolichospermum were observed at beaches at over 50 million cells in the worst observed areas. There were multiple samples collected weekly since the onset of this advisory, just like the other ones, and we're seeing sort of that consistent population, mostly microcystis, you know, across the whole system, especially piling up on our beaches. But what was interesting, and I'm glad Liz captured this because she went out and she got to sample closer to the intake location. And she took a photo here that it doesn't look too bad, especially when you compare it to the other beaches. But this photo represents around 100,000 cells of microcystis, which still exceeds our, you know, recreational guideline. But there's no doubt that microcystis blooms in New Hampshire that we've seen so far do produce microcystins, and it's only you know, a matter of concentration to know how, you know, toxic they could be at any one location. But you know, the question is how much of this is getting through to the finished water. So these microcystis colonies produce microcystin, and the microcystin toxin is very stable. So sometimes even after a bloom is sort of dying out or dead, microcystin, the toxin, can still be around for several days and can easily get through filtration. So I'm back to, you know, what information do we have on these water bodies? Well, again, if you go to our DES lake information mapper, again, you can see what lakes, you know, we have on the map and what information we have on these water bodies. Unfortunately, Arlington Mill Reservoir is not in the Voluntary Lakes Assessment Program. I'm not sure if it ever has been, but at least recent years has not been. The most recent survey that we have is from 1998. Our trophic surveys usually we have 1,000 lakes in New Hampshire, so we usually cycle these every 20 years. So perhaps Arlington Mill is due for another survey soon. But we just can't get to all the lakes every year. So that's why there's such a gap in time and understanding what's going on in the system. And without a volunteer lakes program or, you know, even UNH Lake Slate monitoring program participation, we don't have a lot of data on the water body. We know that it was classified as mesotrophic in 1998. It had a Secchi disc of around 2 meters, which is pretty common, not too bad. But the bottom dissolved oxygen was very low, and that was noteworthy for internal loading for phosphorus. It also has a pretty good flushing rate, and that flushing rate is around, you know, 7 times per year, which is pretty good. However, it's, you know, all kind of moving towards perhaps the intake area. We're not exactly sure how that would work, but we do know that the Arlington Mill bloom was draining down to Wilson Lake, which you can see in the map here. So there can be some downstream effects, but there can also be, you know, sort of that passage through the intake area for these blooms as well. And for what we have on record for the cyanobacteria warning history was there was an event in 2018, and I know that this advisory lasted quite a long time, 66 days, and that got very close to, you know, the end of October when they were considering moving to this water source to be used. So it's very close to these times where you might be thinking about transferring the supply, or, you know, however you guys do that. But, you know, if you are using Canobie Lake and thinking about moving to Arlington at this time of year, it's really important to be monitoring for cyanobacteria. So that brings me to just a few, I'll just, you know, I know I'm running out of time here, so I'll just introduce the Cyanobacteria Monitoring Collaborative, which you can view at cyanos.org. This is an EPA monitoring collaborative with a lot of states. It kind of started in New England and it's now expanded greatly. And this is a great approach because they do have co-ops and SOPs and videos to go through all of the methods that they are suggesting. This is also a tiered approach. So you can kind of choose, what you can do just to be on the lookout, or what, how severe is your situation? What level should you be at? If you're at a system, you've never seen the water turn green, you've never had a report, there's no bloom history, maybe just being on the lookout is okay. Or maybe you want to do a little bit more and, do some of these water quality parameters that are missing in the gaps of time here. Or you could be involved with this cyano monitoring effort with EPA, which just is really collecting water and checking pigments and looking to see if there's a relative change over time. These pigments can be really telling of a cyanobacteria presence, which you would also just want to check with cyanoscope. And that's just using a microscope and looking to see what do you have in the water. Usually using a plankton net helps just to concentrate it, because if you're not seeing anything, that's great, but there might still be cyanobacteria growing in the water. It might just be a good to know approach. And these things, you can also bring samples to DES and we can do, the cyanoscope side for you. And that is a free service from DES. And just so you can see, if you are interested in this approach, what the equipment really looks like, you can again opt into any tier, but basically we're talking about just water tube collectors, plankton nets, brown bottles, and a microscope and a fluorometer if you're interested. And there are nice YouTube videos, are just about 2 to 3 minutes long in most cases, that go over each part of the methods, which is a great way to learn as well. And with that, I will end here, but thank you for your time, and I'm all set.
Thank you, Amanda. I guess we could take 1 or 2 questions. If anybody has one for Amanda, you could unmute yourself and ask.
Hi, I don't have a question necessarily, but I did have a comment about the SONS data from Block Lake. I know there was a question on the units.
We're having a very hard time hearing you. It's all echoey.
Okay, I might just type it in then. How about now? Can you hear me okay?
No.
Okay.
Well, why don't we, if you're not speaking, if you could please mute yourself. We do have one question coming in about risk factors.
Oh, okay. So I didn't really go over toxicity as much for risk factors, because I think that we're going to hear from others about that.
Yeah, we do have a speaker from the EPA that is going to be talking about risk factors. So if that's okay, if we can, we'll just, if you want to wait, and you can ask your question to Leslie D'Anglada. She will be covering that extensively coming up very shortly. Okay, so I don't see any other questions. Well, thank you so much, Amanda. We appreciate it. And next up, we have Katie Foreman from the US EPA, and I will switch her over. Katie? Great. So Katie Foreman is an environmental protection specialist with the US EPA's Office of Groundwater and Drinking Water, and leads the office's work on harmful algal bloom and cyanotoxin issues in drinking water. Katie also serves as a source water quality technical expert in the evaluation of unregulated contaminants and national primary drinking water regulations. Prior to joining the EPA in 2015, Katie led water infrastructure projects with the Oregon Department of Environmental Quality, served for 5 years as a scientific and technical expert on the Chesapeake Bay watershed water quality issues with the EPA's Chesapeake Bay Program Office, and worked for 6 years with the Iowa Department of Natural Resources Watershed Monitoring and Assessment Program, focusing on harmful algal bloom, nutrient, and microbial non-point source pollution. Katie, when you're ready. Thank you.
Thanks so much, Liz, for the introduction. Can everyone hear me? Can you hear me okay, Liz?
Yep, I can hear you and I can see your screen.
Excellent. Well, it is such a pleasure to be with you all today, and thank you to everyone at New Hampshire DES for the warm welcome and the ability to participate here today. I'm going to be participating with a couple more of my colleagues here from EPA. I am with, as Liz mentioned, the Office of Groundwater and Drinking Water at EPA headquarters, and I'm really excited to talk to you a little bit about what we have available to help in developing response plans for cyanotoxins. And hopefully this discussion will really help to set the stage, if you will, for the scenarios we'll be talking about today, and also hopefully be a building block for the other discussions and other presentations we'll see today. So I'm really going to talk about some of the drinking water challenges, although that was covered a little bit earlier by Paul, but I'll also really dive in deep on some of the tools we have for managing the risks of cyanotoxins in drinking water, specifically as it pertains to response planning and being prepared for events before they happen. So I don't have to tell probably this group what the challenges are of cyanotoxins in your source waters and of course your recreational. When we're talking about drinking water quality, as noted before, there are multiple issues around having a bloom in your source water. You could have taste and odor issues. In your finished water, you could have actual health effects from potentially elevated levels of toxins getting through to finished drinking water. And also, as mentioned earlier, you have this increased organic matter in your source water that can lead to disinfection, more disinfection byproduct potential formation, because these blooms can serve as DBP precursors. So, and obviously you're not treating this bloom in a vacuum. You have to meet all your other treatment objectives at the same time: turbidity, removal, disinfection, of course, DBPs as your taste and odor control, as well as corrosion. So understanding there's a lot to balance here. And so with that in mind, in the last probably 5 or 6 years, we put out quite a few resources that I'm hoping I can kind of go over in a lot of detail today, because I think it's really going to help with some of the scenarios we talk about this afternoon in particular, especially as you talk about getting ready and being prepared with a plan for managing cyanotoxins. And I'm not going to go over all of these, but just to say that we have a large array of tools available specifically for managing HABs in drinking water and cyanotoxins in drinking water. And they're all available online. And I have a link here at the bottom and can provide that later as well to get all of this materials. But just hoping that for those of you that may have systems that are vulnerable to HABs, you can put this as a bookmark maybe on your web browser to just know that these are available, and likely questions that you have, others have had as well, and we've really tried to put resources out there to help you manage that. So I'm really going to talk mostly about the top 4 bullets here. These are our biggest sort of resource tools for managing and planning response planning for HABs. I'm going to talk a lot about the recommendations document, which is really sort of the cornerstone, if you will, for a framework to develop preparedness and planning for cyanotoxins in your systems. And then also talk about the management plans and example plans as a way to get started implementing some of the recommendations in that document. My colleague Tom Waters will be discussing water treatment, the document we have on water treatment optimization and other treatment specifics later in this webinar series, so I won't cover that. I'll leave it to Tom to cover that and answer any questions you have on that, but I'll mention it a little bit as it gets into talking about the planning. And then also, of course, communicating risks. This is a big portion of trying to manage cyanotoxins, is understanding the risk and communicating that to the public and other stakeholders, so I'll be discussing that. We also have quite a few fact sheets available. I encourage you to look at our website and just get familiar with the information that's available. And we have, other kinds of media available, such as videos that summarize some of these tools. And we also just want to mention really quickly, we do have a newly released Cyanotoxins Preparedness and Response Toolkit that kind of takes all of this information I'm mentioning here, these resources, and puts it in one spot for both drinking water and recreational waters. So that's also available as well. And I encourage you to take a look at that. But for now, I'm really going to dive into what I think will be helpful in our discussions later today as we talk about developing plans and being ready for blooms. And this recommendations document was one that was published in 2015. And although now it is dated, which is hard to believe, it still is very relevant, and stand as well as a lot of the great background material as well. So if you only have one resource, this is the one to have for being sure you kind of have an idea at the very high level about how to respond to HABs in drinking water. This was released in tandem with our health effects, our health advisories and health effects support documents for microcystins and cylindrous spermopsin as a way to really help out for public water systems and others, how to implement those health advisory numbers. So I'll talk about that in a little bit, but most importantly for a takeaway on this is that within this recommendation document, it provides a framework, and this framework is based on five steps, and it's meant to be risk management in an adaptive framework, and monitoring, treatment, and communication are part of every step of it. And just to give you a sense, and I know everyone has their own flow charts, and I really enjoyed seeing the ones earlier today specific to New Hampshire, but this is one that's really showing more of the framework to give you a sense of the steps. And although a flow chart can't always show cyclical cycles and show that it is in fact an adaptive management loop, I want you all to note that this is adaptive management, that at any point during or before a bloom, you should be in one of these steps. You should be either looking for a bloom, looking for visual signs of a bloom, preparing for a bloom, actively managing a bloom, or going back and determining whether or not you may have another vulnerability that's come up. So it's more of a cycle than it is a linear flow. And the more you kind of get that, I think the easier it is to understand some of these steps. And I'm going to do my best to walk through them in not a lot of detail, but enough to kind of give you a sense, because I think it'll be really helpful as we go through the scenarios later to give you a sense of the framework that at least EPA has out there that you could use to start to think about being ready for a HABs event and preparing for it. So, you know, one thing that might not be obvious, but I want to mention anyway, is that knowing whether you're vulnerable to a HAB is really important, whether your source waters, your surface source water system, and are you going to be vulnerable to having a HAB in your system, and are you going to have to prepare for that? And there are a lot of ways to know whether or not you are vulnerable. I think we just saw some great resources on monitoring information and source waters, but there's lots of other indicators that you could use. Obviously, if you've had a bloom in the past, that is a great indication that you are vulnerable and you'd be put in that category, but there are a lot of other different kinds of information you can use to determine whether you are vulnerable. And then once you determine whether you're vulnerable and you make that characterization, let's say you are, knowing when you are vulnerable and what times of year will be really helpful, because it'll help you kind of walk through the stages of this risk management in a way that makes sense for your specific system. And then of course, step two, being prepared and observing. I just want to note that there's a lot of information on this available in the recommendations document and well as our other materials. So I'm going to go over this very quickly, but there's so much more information that you can use to dig into this. It's a lot more nuanced that I'm going to present here, but I'm happy to answer any questions as well. But of course, preparing is a huge part of having a plan in place. You know, if you have a plan in place, but you don't have the materials necessary or know what your treatment options are during a bloom, you know, you're kind of you're not as prepared as you could be. So these are the kinds of things that, you know, take some time and take some coordination within your, maybe within your public water systems or within your different agencies. But knowing what kind of monitoring information is available, where your labs are, who you can send it to, the turnaround times for that, whether or not you're going to be doing raw, in-plant, or finished water or all monitoring, when you will be doing that monitoring. And of course, the treatment options you have available. What are your treatment adjustments that you could do prior to the bloom? Other mitigation strategies you can use, including alternative sources, blending, changing the depth of your intake, those kinds of things. And develop a plan for, when you'll be doing those kinds of things at what time. When during the bloom situation will you be, looking to make treatment adjustments and those kinds of things. And I can mention, our risk management framework for that. But just being sure that, I think, is going to be, is step really. And having a list of who you're going to communicate with. So your primacy agencies, your local public health officials, others also, I mentioned, really already have networks they can tap into here. If you don't, this is a great opportunity to develop that network of discussing. Let's say you have upstream public water systems that you sort of like earlier in the watershed or upstream in the watershed, and you have communication to kind of get a sense of where the bloom is. That could be helpful. Or you have communication with recreational water quality managers that are managing your source, those kinds of things. Just having that in place and having a strategy for communicating is really important. This here is that you're preparing that. You're preparing those lists, you're getting those phone numbers, you're writing them down, you're figuring out when you're going to do treatment, how, and what's your monitoring scheme, those kinds of things. And then you're really getting into the observation piece, looking for early warning signs. So say you are vulnerable, you're all prepared, you know when you're going to monitor, those kinds of things. Now you're looking for early warning signs. Maybe you have changes in your system. You have changes in the filter runtime, pH, something that is giving you a sense that you maybe have something going on or you're actually seeing a bloom. And any other indicators that might come up in your source water or in your raw water, changes to water quality, those kinds of things. And just, being aware of that. Eye on those, and that's going to be, pretty constant. If you're vulnerable to HABs, it's going to be something you're doing throughout the HAB season daily to see whether or not you need taking more. And I'm going to go over these steps. They are very compressed into one slide. I wanted to be sure I could get through it all, but we can talk about them. If anyone has any questions on any of these different pieces, there's a lot of elaboration in our documents on it. But these are kind of the bulk of it. This is the action-oriented kinds of steps. This is like you have seen a bloom, you are vulnerable, and now you're determining whether or not cyanotoxins are in your raw water. You're determining whether or not it's confirmed in fish water. You're determining whether or not whatever you're doing in terms of in your plant is working in terms of the treatment operations and adjustments. And you're also doing your best to, monitor those treatment, whether or not those are working, and making the active treatment adjustments at the same time. So it's a bit of a adaptive management loop, and hopefully you're making, some real-time decisions and actively doing things in parallel. And then, of course, communicating with partners local at the state and local level, and then also deciding when to inform folks of their risks, especially your consumers. If something has broken through, cyanotoxins have broken through to your finished water, really thinking about when and how you're going to communicate with the public. When are you going to make those communication press releases? If you will end up issuing a do not drink order based on whatever level of cyanotoxins are in finished water, how you're going to do that, what's the steps that you're going to take. And this really is these steps 3 through 5. You're likely, if you have an active bloom and it's breakthrough to finished water, you'll likely be in these steps for a while because, until it's sort of resolved in raw water, you'll want to continue to be looking and monitoring raw water and making treatment adjustments as necessary. And hopefully if you have some in-plant monitoring as well, doing that actively and adaptively. So that might be a little abstract, these steps 1 through 5 in that flow chart, that we noted that when we developed that recommendations document and thought it might be helpful for public water systems to have a template to use that incorporates this 5-step process. So again, the best way to be ready for a bloom is to have a plan in place. And this plan is a template that we've developed that is really easily adjustable for your own situations, and also has that framework set in place, that steps 1 through 5, where you can put in your own action levels and your own system-specific information and actions that you plan to take, including any sort of trigger levels for when and how you just monitoring treatment or when you communicate with the public. And what I want to note about these is that we also have 5 example plans in this document as well, so you can see how system-specific these plans are. It is not sort of just, everyone's this, you kind of like everything's the same. It's just sort of stamped and everything looks the same. We have systems from New York and Oregon and here in Ohio that show different ways of managing risks using this 5-step framework that works for their particular system. It's really great to see a variety of risk management and how they're implemented. And there's a lot of ability to make things your own and be very specific to your own needs. So I encourage you to take a look at those example plans. And I also just encourage you to know that you don't have to start with a blank page. That can be hard when developing a plan. This is an option to download this and update it with, phone numbers, communication information, monitoring, treatment information, just to be really ready for an event. The other thing I was going to mention is that within those plans, you're going to be really leaning on a couple of our resources. You'll be leaning on that recommendations document when you're developing your plan, that risk framework, obviously. You'll be leaning on the treatment optimization document that I noted earlier and that my colleague Tom Waters will discuss. And then you'll also be looking at this risk communication toolbox. I feel like this is a great, some great content for communicating the risk to the public, including templates and things that you don't have to spend a lot of time developing on your own. You can, it's meant to be adaptable to different situations, but has language that is known to be effective in communicating with the public about the risks, is accurate, has good information behind it, and is something you can just use quickly. You don't have to spend your time, let's say you are in an emergency situation, you don't have to spend your time developing communication products. There's something ready for you to use and ready for you to communicate with. So I highly recommend taking a look at this and seeing what's available so you can think about how you might want to use it and adapt it for your own communities. And I want to note that the target audience is the public. So this is the point, is to communicate with the public. This isn't for internal partners. This isn't for those kinds of situations. This is specifically for risk communication with the public. And it's available in English and Spanish. I have the links here. Highly recommend taking a look at it. And I want to note something about these in particular before I get to some of the action levels that may be used with these materials, is that these aren't meant to be this toolbox is not a communication plan. They are communication materials. So you will have to decide based on your particular local level or state information about when and how you're going to communicate with the public. This is just materials to help you do that. Determining when and how you're going to communicate with the public is an important first step, and as mentioned, that's in the communication part of the management plan development. But this is going to be great information to use when you are communicating with the public. And this sort of gets me to the discussion I've been sort of talking about a little bit about action levels, or when and how to communicate with the public, and specifically when you're talking about potentially do not drink situations. Within this toolbox, we have press releases, we have drinking water advisory texts as necessary, and what we use are REPA National Drinking Water Health Advisory levels. And I believe this was mentioned a little bit earlier today, but I'm just going to go over it really quickly. We do have, as you all know, that cyanotoxins are not regulated as a national primary drinking water regulation. They are an unregulated contaminant, but we do have these federal health advisory levels that were released in 2015 for both microsystems and cylinders per mopson. And as many of you probably know, obviously, and you see here that we have 2 different types of toxins with 2 different populations and 2 different levels. So we've got 4 total to try to communicate when we're trying to talk about risk. And that can be complicated, but is not impossible. And there are many great examples of how that has been done and well understood by the public. And I just want to note that my colleague Leslie Denglada is going to be talking right after me here on health effects, and specifically not necessarily going over the health advisory numbers, but talking about what is behind that. What does it mean that we have these advisories? What are the health effects of these cyanotoxins, just so you know that information. And we can talk about these more later if there are more questions about these health advisory numbers, but I just want to make it known that we use in all of our materials this number and these sort of advisory levels, but they can be adapted as necessary for any kind of level that either you locally come up with or is required by any kind of state or an other agency. So I just want to make that note. And as I mentioned before, I'm just going to kind of go through a little bit on this because I think there's a lot in this toolbox, and it can be very useful for the communications piece of your plan. And as I mentioned, there's several different levels to this. We have drinking water advisory templates, press releases. We also have social media and text alerts that are really ready to be adapted and just sent out, which I think can be really helpful. We've heard in drinking water emergencies that public water systems are spending a lot of time with risk communication. They're trying to develop language, and this is something you don't have to do. If you want to use this information, you don't have to do that on the fly. It's already ready. And especially if you are in the situation where you're issuing do not drink, especially you have levels that have broken through to finished water. This is going to help you execute that do not drink if you decide to do that, and will help you get the idea of the kind of language that can be used and is recommended for these different levels and different scenarios. For instance, if the levels over the exposure level, health advisory level for everyone versus just vulnerable populations, and then we have additional language for when that advisory is lifted. So I'm spending a little time on this just to be sure that you know that there's a lot of materials there. I think it can get lost a little bit in our website. And again, we also have co-branding available. You can see there in the bottom kind of screenshot there of that fact sheet. You can add your own branding if you wanted to do that, or just use it as is. And something I'm going to mention, I think we're going to be talking about a little later, is also the importance, I think, of communicating in multiple ways. There are a lot of different ways that people learn, and communicating in lots of different ways to try to meet all audiences, I think, is really a very important thing, and trying to be as minimally complicated as possible in that communication. So we've developed some graphics that maybe will help as you're trying to communicate with the public on different scenarios for, whether if you do decide that you're issuing a do not drink, what does that mean? What can and what should customers use the water for and not use the water for? And of course, in every scenario where it's broken through to finished water, there's a do not boil, and hopefully you can see that in some of these actionable graphics. But just want to be sure folks saw that and get a little familiar with it if you're interested in using them. Of course, you can use anything to communicate. We're just trying to give some options. So with that, I want to just provide my contact information, and also want to provide a couple links to the Cyanotoxins and Drinking Water website, which includes all the materials I just discussed today, and then also our broader CyanoHABs website. And just for good measure, another link to that sort of comprehensive toolkit we have for preparedness and response. And really looking forward to answering any questions you all have, and seeing if we can put some of these ideas into practice later this afternoon. Thanks for your time.
Thank you so much, Katie. If anybody has questions for Katie, feel free to unmute yourself and ask your question. Okay, I don't think we have any questions at this point. Doesn't look like it. Well, thank you so much, Katie. We really appreciate your presentation. Next up, we have Leslie D'Anglada, and I will make Leslie the presenter.
Hi, Liz, can you hear me?
Hi, Leslie. Yes, we can. And I just gave you the controls to show your presentation.
Wonderful. I'm so happy to be in the computer audio today.
Yes, thank you. I saw that. So I will introduce Leslie now. Dr. Leslie D'Anglada is a senior microbiologist with the United States Environmental Protection Agency. She serves as the harmful algal blooms lead for the Office of Science and Technology and Office of Water. Leslie is the manager of the EPA Drinking Water Health Advisories for cyanotoxins and co-manager of the Recommended Recreational Water Quality Criteria and Swimming Advisories for cyanotoxins. Leslie represents EPA in national and international committees and advisory groups, such as the Interagency Working Group for Harmful Algal Blooms, Hypoxia Research and Control Act, the National HABs Committee, and the World Health Organization's Water Quality and Health Technical Advisory Group. Whenever you're ready, Leslie.
All right, can you hear me now?
Yes, yes.
Wonderful. I think I already moved the slide. So thank you, Liz, for inviting me and for the opportunity to present today on the possible health risks from exposure to cyanotoxins in drinking water. So we had a question already about the risks. So the purpose of this presentation is to talk about them. Even though I just moved to the next slide. We mentioned the health advisories. I will not be talking about the health advisories today, but I will be talking about the possible health effects when ingesting 3 particular common cyanotoxins in the US. And then I will provide an overview of EPA's approach to address harmful algal bloom in freshwater systems, so you know what EPA is doing. All right, so today I will not be talking in depth about what are harmful algal blooms. I think Amanda did a very good job. Oh, can you still see the slides?
That one, and then it looks like it's going back to the non-presentation view. That is the presentation view.
Okay.
Mm-hmm.
we're okay then. All right.
Yep.
So I just want to say that in freshwater systems, cyanobacteria are the most common type of algae. Although they are bacteria and not properly algae. And since I'm a microbiologist, I have to be very specific on that. And so we hear about the red tides and the marine side. So these are 2 different types of organisms that produce harmful algal blooms. And we have experienced in the past several years several major cyanobacterial harmful algal blooms events in surface waters, affecting mostly recreational waters, but we have also seen drinking water systems affected by cyanotoxins across the US. Here, I'm introducing the One Health concept, as HABs are as a good example of a One Health problem. I don't know why my computer keeps doing that, so I hope you still can see the slides. And we know that people can be affected by harmful algal blooms, that animals can be affected, and that harmful algal blooms also affect the environment, and that's why it's a One Health problem. And effects, adverse effects on people, animals, and the water quality also have economic consequences. So harmful algal blooms can have a serious impact, not only to health, but also to the economy. The illnesses from exposure to cyanotoxins vary depending on the toxin you are exposed to, how you are exposed, and the dose of the toxins you are exposed to. Those are very important issues you need to keep in mind because these toxins react in different ways. People react differently to toxins and contaminants, and the dose of the toxins could also vary and have an impact on the health effects. There are several ways people can be exposed to cyanobacteria cells. So you can be exposed through the skin to cyanobacteria cells itself and to the toxins while you are recreating in toxin-contaminated water. You can breathe toxins in aerosols, and aerosols are tiny airborne droplets. It's not that the microcystin or cyanidospermopsin can evaporate. They can't. So it's these mist or droplets that could be found in windblown sea spray or windblown water spray. You can ingest cyanobacteria and their toxins incidentally while recreating, or you can ingest cyanotoxins in drinking water or contaminated water. Another example of a root exposure is via intravenous exposure, which is less common, but it had happened in dialysis clinics with contaminated water. Next slide. All right. So We don't have adequate toxicity data for some routes of exposure to determine what are the health risks and to develop health advisories and guided values. For example, we don't have adequate toxicity data for anatoxinA. So we have not been able to develop health advisories for anatoxinA. And we don't have toxicity data for inhalation to aerolized toxins. So we don't have any recreational criteria for inhalation to aerosolized toxins. But we do have, for some toxins, good oral route of exposure toxicity data, and those are the ones that we have developed risk assessments. And so when we do risk assessment to determine that possible health effects, we use dose response data from toxicity studies to develop that reference dose. So in the following slides, I will discuss a summary of those acute, subchronic, and chronic effects. So we determine what are going to be your health effects if you're exposed to one day to a toxin. That is an acute effect. We do subchronic or short term, which is 7 to 10, 14 days, 2 weeks, if you're exposed to those toxins during that time. And we can also develop health advisories or determine what are your risks for chronic effects, which is a long-term effect, which are you are exposed more than 30 days to persistently to a contaminant. So the health advisories that we develop are based on information on short-term effects. So we are saying that you are exposed during a short-term exposure, right, to that specific toxin, because we know that blooms tends to be sporadic, right? So they tend to be more during certain months, they come and go. So the exposure that you are having to those toxins in drinking water or during recreational activities are not long-term, are just short exposures. So the health advisories are based on short-term exposures. So let's start with microcystin. Most of the toxicological data on the effects of microcystin is limited to the LR congener, and up to now we know of around 200 different congeners or variations of microcystins. Therefore, the evaluation of health effects from exposure to microcystin is based on that particular congener. Microcystin, when it's ingested in water or in food, so we're talking about oral exposure, they are transported using receptors from the intestinal tract to the blood. So they pass through our intestinal tract, they are in the blood, they circulate through our body, and they attach to proteins in the liver. So the liver is a magnet for microcystins. Microcystins attach to the liver and they start doing the changes and damages in the liver. We see that either by sonograms or we see that through blood testing. The limited data that we have on human data have observed that there is damage in the liver, but the data that we get is not adequate for us to use. So we use laboratory data. So there have been several studies that have been done, and the studies demonstrate that exposure to the microcystin could affect the liver, but also could affect the gastrointestinal system and the reproductive system, especially the male reproductive system. But we know that for the lower dose, the liver is the organ that has the most effects. Studies that evaluated the long-term effects, we found 2 studies, but the results are inconclusive as the studies were limited in the design, how they were done, and also on the data that it was reported. So we cannot say for sure what is going to be the health effects if you're exposed to microcystin for a long time. We also evaluate, and those previous effects that I mentioned are non-cancer effects. So we also, when we do risk assessments, we evaluate what are the carcinogenicity of particular contaminants, in this case, microcystin. We found several human epidemiological studies that were done in China that reported an association between drinking water with microcystins and liver and colon cancer. However, the studies are limited by their study design. they have poor measures of exposure, potential co-exposure to contaminants like chemicals that could cause liver or colon cancer. And the researchers couldn't control for known liver and colorectal risk factors. So when we follow our EPA guidelines on determining if a contaminant is carcinogenic, we determine that the data that we have for microcystin is inadequate evidence to determine the carcinogenicity of microcystin at this moment. The next cyanotoxin that I will be talking today is cylindrospermopsin. This is a tricky cyanotoxins because cyanotoxin, usually the toxins are retained within the cell unless they're dead, they die or they break down. But cylindrospermopsin tends to be released in the water even when the cell is intact. So you can find half of the toxin in the water and half inside the cell, which is problematic. Although the mechanism of toxicity of cylindrospermopsin had not been fully characterized, we don't know everything about how cylindrospermopsin causes toxicity. We know that once it's ingested, cylindrospermopsin is absorbed from, again, the gastrointestinal tract, goes in the blood, and circulate through the body, and like microcystins, attached to the liver. However, the mechanism of attachment to the organ is different. So it tends to float between the liver and the kidneys. And we have observed that at lower levels or concentrations, it could affect more the kidney than the liver, but it could affect both. So it's a liver, it's a hepatotoxins, but the most of the damage is on the kidneys. Long-term toxicity studies have not been done, so we are not sure what is going to be the long-term effect of cylindrospermopsin. Several laboratory studies have found the potential damage to DNA by cylindrospermopsin. However, there is no up-to-date bioassay or a long-term study to evaluate the cancer potential of cylindrospermopsin, and those are two requirements that we need in order to determine the carcinogenicity of a contaminant. So at this moment, we cannot determine if cylindrospermopsin is carcinogenic. All right. Sorry, I lost my cursor. Oh. All right. The last toxin I will be discussing today is anotoxin A. Anotoxin A has been associated to the death of animals, especially dogs, in toxin-contaminated water. However, when we try to link the exposure to the effect, it's usually difficult because anotoxin A undergoes rapid degradation in surface water in the presence of sunlight. So it makes hard to detect the cause and effect during dogs and animal poisoning events. We know that once anotoxin A is ingested and is distributed in the blood, just like microcystin and cylindrospermopsin, anotoxin A attached to the neuromuscular nicotinic receptors that are located in the brain. And anotoxin affects the central nervous system. So anotoxin A is a neurotoxin. It's different than cylindro and microcystin. There are no human data, no case reports of oral toxicity of anotoxin A. And there are two short-term studies, laboratory studies done that found changes in the blood and enzyme levels. But we have no toxicity effects caused by anotoxin A. Therefore, we were not able to determine where is that reference dose or that dose that will protect you if you are exposed to anotoxin A in drinking water. That dose is the one that we use to develop health advisories. Up to date, there have not been no chronic studies, so we don't know what is the chronic effect.
Excuse me, Leslie, where your presentation switched back to non-presentation mode. So we're at your first Okay, now we're back. Yep.
I know. It's it's something in my computer. I'm so
Nope, now we're back. Now we're good. Thank you. I just wanted to let you know.
Liz, you should know that with me anything could happen. I'm a
know.
box full of surprises. All right, so we don't know anything about the carcinogenicity of anatoxin A. There's no information on carcinogenic information. So I'm just going here. I don't know. Here we go. Okay, so here's a summary of adverse health effects. Again, human health effects from exposure in drinking water to microcystin are changes and or damage to the liver, and exposure to cyanidospermopsin are changes or damages to the kidneys. Now, these changes, as I mentioned, are going to depend on the dose or the levels of toxins you're exposed to, or how long or the total time that you've been exposed to that toxin, and of course the individual person characteristics. So for example, your age, immune system status, and previous health conditions, such as people with liver and kidney conditions or disease. The main toxic effect of anatoxin A is acute neurotoxicity. From the laboratory studies that have been done in animals, they have found that it could, cause respiratory paralysis just in seconds, and the longer time was 2 minutes. However, those studies, again, those are what we call dose studies. They are done to determine what are the doses that we can use in toxicity studies with animals. But again, we don't have toxicity data for us to determine what is the health risk or health advisories. And so let me go back. We have not developed health advisories for anatoxin A. And I think I should mention this, that health advisories, they are not regulations. Katie mentioned that, and they change as new information becomes available. So we continue evaluating, and we are planning to continue evaluating health data for anatoxin A to determine, to try to develop a health advisory. So these are the toxicity research needs that we have identified. I'm not going to go over them. They're too long. But as I mentioned, we need information on anatoxin A, and we need information on other exposures, such as inhalation. Very briefly, I included here other adverse effects from exposure to cyanotoxins. We know that cyanotoxins have been associated with the death, as I mentioned, of wildlife and domestic animals. And the animals are exposed to either drinking water, or I don't know why, dogs love eating the scums and the algal biomass. And if anatoxin is there, the death is very, very rapid. Cyanotoxins can also be found in fish and shellfish, freshwater. They accumulate, and they can bioaccumulate in the organs and tissues. And we know that they are concentrated more in the liver, because, as I mentioned, it's a magnet for toxins, and the hepatopancreas of toxins of fish. I'm sorry. So we recommend not to eat those organs when you are eating fish that could have been in a bloom. Cyanobacteria cells and toxins have also been found in irrigation water, and they could move through the roots and migrate to the grains and fruits and be in the plants and crops. We have very little data, but we have studies that have found that association as well. So we continue evaluating that data to determine in the future what is the health risk if you're exposed to crops and plants that have been irrigated with toxin contaminated water. As I mentioned at the beginning, we have a One Health approach to deal with harmful algal blooms in EPA. And we develop guidance and recommendations, risk communications. We conduct assessments and monitorings. We do outreach, and we create partnerships. And of course, we do also research and development and provide technical guidance. So you met Katie. Katie have done a lot of work on the outreach and guidance and development of recommendations. You're going to talk and hear from Tom Waters, who have provided a lot of information on providing guidance on optimization and providing technical support as well. And so I'm just going to go over very briefly on the things that we have done. As I mentioned, we don't have any federal regulations for cyanotoxins, but we have developed health advisories for 2 toxins, and we have provided a supplemental and technical guidance documents that Katie mentioned. We do assessments both in drinking water with the UCMR and also in surface water and rivers, streams, estuaries with the National Aquatic Research Surveys. We develop outreach and communications materials. We have several websites. We have a monthly newsletter. We conduct webinars, and we have done regional workshops, both for drinking and recreational waters and issues related to HABs. And we create partnerships as well with work groups and projects with regional offices activities, as well as we do research. This is my contact information. That's the last slide. I want to show that picture that is Pandora. Pandora is one of 6 ducks that in the summer of 2018 were exposed to a microcystis bloom in Martin County, Florida. Pandora was very sick, but thanks to the prompt veterinary care, 5 of the 6 ducks survived, being Pandora one of them. And since then, Pandora has been very actively advocating for clean water in Florida. So that's all for my presentation today. And I can take some questions now.
Thank you, Leslie. If anybody has any questions for Leslie, please feel free to unmute yourself and ask your question. I don't think we have any questions at this time. Thank you so much, Leslie.
Thank you, Liz.
Next up, we have Lacey Gores-Priest, and I will make her. Okay. Lacey Gores-Priest is the water quality supervisor for the city of Salem, Oregon. She oversees drinking water compliance, including backflow prevention, regulatory compliance sampling, and watershed monitoring and compliance. During 2016 and 2017, Lacey served as chair of the Pacific Northwest section of the American Water Works Association. Welcome, Lacey.
Good morning. Can you see my screen?
Yes, we can.
Perfect. Okay, great. Thanks for having me this morning. Coming to all the way from the West Coast. It's pretty early over here. But I'm excited to share our story with you from a utility perspective that actually went through a algal toxin advisory experience. It's very timely considering we're near Halloween, because it's a very scary story that I need to share to help other utilities best prepare for this type of advisory, because it can happen to you. So today we're going to talk a little bit about Salem's water system, so you have a better understanding of our treatment process, the customers that we serve. I'll do a brief overview of our watershed monitoring program. I'll cover a little bit of algal toxins, although the other presenters today have already provided a great deal of information on that. But my main focus today will be talking about our 2018 Drinking Water Advisory, the challenges that came along with that advisory, and just some good information on communication and outreach anytime you're interacting with your water customers regarding a challenge in drinking water quality. So the city of Salem here in Oregon gets our drinking water from the North Santiam River. It comes from the foothills of the Cascade Range. We are impacted by 2 Army Corps of Engineer dams, Detroit Dam and Big Cliff Dam. Downstream of those dams, 26 river miles, is our water treatment facility called the Guerin Island Water Treatment Facility. We serve just shy of 200,000 water customers. We provide water to 3 wholesale customers, Suburban East Salem Water District, City of Turner, and the Orchard Heights Water Association. The North Santiam River is really our only drinking water source. We do have 4 aquifer storage and recovery wells that we can utilize, but that is treated water from the North Santiam River that we inject underground in a naturally existing aquifer and then recover for supplemental or emergency water. And it only provides really less than half of what our base demand is, so it's definitely not enough water to meet our entire demand during the winter months. So an overview of our water system in 2018. Here is number 1 is the North Santiam River. This is our intake to our drinking water facility. We call it the middle intake. We do have a pretreatment capability that was put in post 1996 flooding that occurred. But our primary treatment is filtration, which is slow sand filtration. So we have 6 2.5 acre slow sand filters that use biological filtration to treat the raw water coming into the plant. We then generate sodium hypochlorite on site. We do fluoridate our water using fluorosilicic acid, and then also we add soda ash to our water for corrosion control. Finally, finished water is delivered into town into 2 large transmission mains. So the overall goal of our watershed program, our watershed monitoring program, is to protect the city of Salem's drinking water by providing advance notice of threats. So those threats could be algal activity, wildfire. Our river runs alongside a major transportation corridor, so we often will see accidents or trucks in the river, or even gasoline, oil spills that we respond to. We have a tremendous amount of baseline data that we collect that just helps us better understand the water that is in our watershed and helps us best operate our water treatment facility. We've been collecting data really since about 2009. We initially were collecting nutrient data, and then we had an event in 2009 where we had 2 filter clogging types of algae that severely impacted our ability to produce drinking water. And we actually had to go into a stage 2 water curtailment, voluntary curtailment for our water customers. And so we really amplified our watershed monitoring program after that event, where we began collecting the algae ID and enumeration samples, general water chemistry, as well as in 2013, we did start monitoring for cyanotoxins after we did notice the presence of cyanobacteria, obviously, in our raw water up there, so we could get a better handle of what we were seeing in the watershed. So those are just a couple of pictures of different blooms that we've seen in Detroit Reservoir. So we've already covered this this morning, but really, what are cyanotoxins? They're produced by several different species of cyanobacteria. Even if there are toxin-producing cyanobacteria present, it doesn't mean that a toxin will be produced, and it doesn't mean that the scale is the same. So you can have a really large algal bloom with very little toxin produced, or you can have a very small bloom with a lot of toxin produced. It makes it difficult to know that just because you are seeing a bloom present, that you're also going to see that toxin present at significant levels. Some species of cyanobacteria can produce multiple types and variants of the cyanotoxins. And then we've already covered the types of health effects that can vary from skin rash to death. So wide range of implications when exposed to these types of toxins. So on the picture there, you'll see that this was the beginning of the algal bloom that started our drinking water advisory. So you can see we had a very dense green bloom in one of the arms that feeds into Detroit Reservoir, which is not unlike blooms that we typically see in May. This is pretty common that we see blooms occur in these arms and ultimately will make their way to the dam and be released downstream towards our intake facility. We've also talked this morning about the cyanotoxin guidance values. So these are the EPA guidance values, and what I will tell you is that it is complicated to communicate this information to the public. You're dealing with some different types of cyanotoxins. You're dealing with a 10-day health advisory level, which is hard to communicate to the public. You're dealing with different levels set for adults versus vulnerable population for each of the cyanotoxins. Plus, there's recreational advisories that can be issued in the water bodies that serve your drinking water sources. And then there's dog guidance as well. So it's a lot of information, a lot of different data points to convey to the public and help them understand what is potentially happening in their source water for their drinking water. So in 2018, on May 29th in 2018, the city of Salem issued a public health advisory for the vulnerable population, which included infants, young children, and other vulnerable individuals. And it not only applied to the city of Salem, but also our 3 wholesale customers. So the city of Turner, Suburban East Salem Water District, and the Orchard Heights Water Association. So we had detections of microcystin that made it through our treatment and entered our drinking water during that time. At that time, I think it's important to note, because earlier in the conversation this morning, we've talked about labs. The city of Salem at that point was utilizing outside labs for analysis of both the algae ID and enumeration samples. So what type of algae was present at what levels, but also the cyanotoxin analysis was done at an outside lab in Ohio. So we were not only running tests for the ELISA methodology, but we were also using LC-MS-MS. So we were getting 2 different types of data reported in, and there was a lab delay in getting that data back to us, which makes it very hard to be making operational decisions when you're using data that is, 2 to 3 days old. We had a fantastic lab that we worked with, and they turned around the data as quickly as possible. But there are just things that you can't avoid, such as, shipping time, data collection, shipping time, the arrival of the samples to the lab, the preparation of the samples, and then the analysis of the samples. And so another really difficult communication piece was the data delay. It also happened to be happening over Memorial Day weekend, and the city faced a great deal of criticism that we were waiting for data, that we weren't, we didn't want to impact our 3-day weekend. And so we didn't issue the advisory till the Tuesday after Memorial Day weekend. Which there was nothing further from the truth, but we were waiting for, sample results to come in, confirmation sample results to come in. So again, a lot of speculation and criticism coming in from our customers related to that. So an overall kind of timeline of events. We started noticing the visual indication of a bloom on Detroit Reservoir on May 8th. We went back up on May 10th, noticed that the conditions had improved. But by the time we went back up on May 14th, the bloom was back full in scale. We increased our sample frequency. On May 23rd, there was a recreational advisory that was issued for Detroit Reservoir. On May 24th, we began making treatment operations, changing in our treatment to be able to compensate for the changing conditions that we were seeing in the reservoir. We began to collaborate with the Oregon Health Authority, who is our regulatory agency. We were also reminded of the 10-day exposure limit, or cushion, which is what it was referred to, which that was a very difficult communication piece. That 10-day exposure piece was something that the public did not, it did not resonate well with the public. They didn't understand how it worked, and it didn't make a difference that there was actually a 10-day kind of threshold for exposure. So we can talk more about that later, but something just to keep in your back pocket if you were to ever experience this type of event. We did activate our backup wells. Then we suffered a large transmission break, which impacted our ability to utilize groundwater instead of surface water at our treatment facility. We had crews that were working over the weekend to repair our leak on our 54-inch transmission main, also waiting for sample results to come back in. On May 29th, we had a conference call with EPA and the Oregon Health Authority. Although we were not at the 10-day threshold level yet, we went ahead and made the decision that we were going to go ahead and issue that health advisory for the vulnerable population, and we issued it later that afternoon. On June 2nd, we were able to lift that advisory after we had 2 good days of data that were below the health advisory level. But unfortunately, on June 6th, we had to reissue the advisory after another result came in that was above the health advisory level. Ultimately, we stayed in the advisory until July 3rd. And the reason we did that is it was self-imposed, but we had engineers working round the clock to add powdered activated carbon to our treatment facility because we weren't confident in the existing slow sand filters to be able to handle any additional input coming in from the raw water. So we added in that temporary change in treatment. Once that was online, we felt as though our treatment would be successful at removal. And so we went ahead and lifted that advisory. As you can imagine, it was a huge, huge deal here in the state of Oregon. Of course, making the front page of our newspaper, the Statesman Journal. You know, the last thing as the water quality supervisor that you want to see is a caption of the front page that says the drinking water is fouled. And so, as you can imagine, we were bombarded with phone calls, emails. We had our utility billing staff taking phone calls, our dispatch center taking phone calls. We had to bring in other staff to field phone calls. I know that the Oregon Health Authority was bombarded as well because we had never had to issue a drinking water advisory for this in the state of Oregon previously. In fact, there, you know, all we had were these health advisory levels, but you know, no actual regulations for the state of Oregon to dictate kind of how we would move through this process. And so, you know, it was a lot of firsts, and I will tell you that it was something that I hope to never encounter again. You know, you think about drinking water and how it impacts your customers, and you know, we were receiving phone calls, you know, for things like, you know, hey, I have a pet lizard. What kind of bottled water should I give my pet lizard? All the way up to calls from our hospital saying, hey, we have surgery scheduled. Can we, you know, are we safe to use the medical supplies that have been laundered in your city of Salem water? Are we safe to do dental procedures? And we didn't have the answers for those questions. And so we were working alongside the Oregon Health Authority and the EPA, and also leaned out to Toledo, Ohio, who had experienced a microcystin event back in 2014 to try to get information out to the public. So I know there was a lot of frustrations because we didn't have a lot of FAQs or information on our website right away because we were still trying to gather that information to put it out to the public. So that was another piece of communications that we hope to improve on should we ever have to go into another advisory. So of the 36 days of the water crisis, we were on the front page of our newspaper for 25 of those days. So it was a long issued advisory, a lot of publicity, as you can imagine. We did do water distribution for homebound folks. We did do water distribution sites that we had borrowed distribution trailers from other utilities through our Warwarn partners, and we were providing drinking water. And as a reminder, you know, this was just for vulnerable population. This was not an all population advisory. So it was also very difficult to explain to people whether or not they fell into that vulnerable population category. A lot of questions about, should I not? And so trying to provide good information for our customers to make good decisions on behalf of themselves and their families on whether or not they should participate in the advisory or not. So again, another difficult piece to the communication strategy for this type of advisory. So after the issuance of our advisory, the state of Oregon, or Oregon Health Authority, drafted temporary regulations very quickly, within a matter, I think, of 3 or 4 weeks, which is record setting. And so what it did is it set health advisory levels for total microsystems and cylindrospermopsin. The levels matched with the health advisory levels for the from the EPA were. And then it gave us a little bit of more timeline. So when you didn't exceed a trigger, in the raw water entering your facility, it really bumped up the timeline and monitoring and public notification requirements needed for utilities that could be impacted by a bloom type situation. And it imposed a regulatory season of May 1st through October 31st for certain water suppliers that are surface water supplier, that get their water from a surface water source, that have either been known to be impacted by HABs or have had the presence of cyanotoxins in their water previously. So these temporary regulations were formally adopted, and now the state of Oregon does have regulations for ongoing regulations for both total microsystems and cylindrospermopsin. So some different challenges that we faced related to this. Critical customer notification. So, depending, it could be a harmful algal bloom, it could be, a boil water notice. I mean, there's all sorts of different ways in things that could affect your customer base. So knowing who your critical customers are and how to notify them in the event of emergency. Reaching out to dialysis centers, hospitals, clinics, daycare providers, the school district, elderly care facilities, just all your variety of critical customers. Those folks that are food processors, the city of Salem has a lot of food processors that rely on our product to make their product. So reaching out to those folks, for every day that they're not utilizing water, that's costing them money. So it has a huge economic impact to those customers. We already talked briefly about the FAQs for customers. So it was a full range of questions, comments from our customers, everything to my chicken won't lay eggs now that it's drinking your water, to, again, can I, my dog died, what should I do? Or, I'm not feeling well, what should I do? So, a huge range of information that needs to be readily available for your customers so that they can digest the information and then also make good decisions for themselves and their family. We did get some assistance from the Oregon Emergency Management, State of Oregon Emergency Management. They sent out an alert, which actually backfired. It was supposed to be an alert that went to the Salem area that just advised that there was a drinking water advisory. And instead it went to areas outside of the city of Salem, also including the city of Salem, but the message that was sent was civil emergency in this area until 11:28 p.m. prepare for action. So as you can imagine, that created a significant a volume of calls and questions about what that meant, but it actually was just an error that occurred. But we didn't need any errors at this point. We were already kind of fully thrown into a communications crisis. So that didn't help the situation. Having two advisories back to back, your water's fine, but now it's not fine. So that really impacted consumer confidence in our drinking water because we had those two advisories that were back to back. It's confusing messaging when you say the water's safe for us, because it was safe. Our data showed that it was safe, but we were still under advisory. And that was because of our decision locally to make sure that we had treatment in place additional treatment in place that could help mitigate any additional cyanotoxins that were present in the drinking water, in the raw water. So that was a confusing message to try to deliver as well. That 10-day exposure, again, very difficult for us to communicate to our customers. They did not care about a 10-day exposure. You know, they felt like if they had one glass of water during that day, that meant they were impacted. And so again, super technical information to try to explain. The vulnerable population and all population language, you know, trying to help our customers determine whether or not they were part of the vulnerable population category. We spoke about the data delay, so getting that data back, you know, a day or two later. So we were making operational decisions based off of, you know, delayed data. And then we also had, you know, at that time we did, you know, we were getting both information lab results in from both the ELISA methodology and the LCMS-MS methodology. And so deciding on, you know, which types of, you know, which method were we going to be basing our decision making on. And so that was a significant challenge as well. Communication looking to the future. In the previous presentations, you've seen some things look like similar to this, where you kind of have the red scale all the way up to the green scale. So this is the City of Salem's version of that, where during algal season, we keep this on our website. We let our customers know kind of which category we are in based off of what we see. Depending on what we see, it triggers different treatment operations. And so we now have a full-fledged watershed monitoring program that is based off of a bunch of different thresholds upstream from our treatment facility. So we are hopefully way ahead of the game to catch anything that's being produced up in the watershed and potentially make its way down to the facility. But by the time it would ever get to the facility, we have basically all of the tools in our treatment toolbox up and running, so we're able to handle anything that comes at us. You know, putting information in our annual water quality report, keeping up-to-date information on the city website. That was kind of a big takeaway for us. The city of Salem, you know, we were kind of old school before this happened. As far as, you know, we sent out press releases. That's how we communicated, you know, and we had, you know, one PIO that initially was trying to take this on as we started, and we quickly learned that is not how people communicate. Social media is where it is at, and you need to have people on social media fast. Social media can be your friend, but it can also be your foe, and the amount of disinformation that starts and can spread like wildfire in a matter of minutes. And so you have to have staff on there putting out those small fires, resharing good information so that your customers are getting access to good information. Trying to communicate about our monitoring schedule, utilizing our community alert system if needed. We had a big campaign following this advisory to make sure that people were signed up for community alerts so that they would get access to information quickly if there was indeed another advisory that had to be issued. Enhanced communication with our key critical customers, so making sure we have good communication either through via email or phone numbers or whatever we need to do to be able to reach out to those customers that use our water for any sort of critical means. Trying to produce information that's easily accessible and the data is understandable for our customers. They feel good about what they're seeing. They have a good understanding. Keeping information available on social media so that folks are sharing that information and spreading good information. And then talking about the treatment changes that we've implemented to further add protection to the drinking water process. So again, how to communicate. You know, we initially tried press releases. We had the one PIO that started this off for our Public Works Department. We then realized that that was not enough. We ended up having to start a joint information center where we had folks coming in from the county. We had folks coming in from police, fire to help us with the public information side of the process. And then we had staff literally just dedicated to social media, people just monitoring Facebook, monitoring Twitter, you know, putting links out to our website because we found that that was how a lot of misinformation was being shared. We did our fair share of press conferences, putting information on the website, putting information out through our community alert system, and then also utilizing the Office of Emergency Management. So looking ahead, things will take forward with us should we ever, need to go through this process again. The Joint Information Center was found to be super useful. If you're a utility that's not super large, your treatment operators or your water quality staff, they're busy out trying to fix the problem. They're out sampling, they're working with labs. You need people on staff or people that you can bring in to handle the communication piece for you. Because as the frontline staff that's working on the issue, you just don't have the time to do that. Make sure that you're talking to your critical customers early and often so that they feel like they are valued and that they are getting the most current up-to-date information so they can make their best business decisions on their end. Making sure that you have a public information officer that understands this regulation and is able to convey that information in a really succinct and informative way to your customers. Make sure your management teams, your boards, your commissions, or whoever you work for know that this is a possibility and what your plan is if this happens. They need to be your ally. Use your trusted communication partners. There's a lot of people that following this event and during this event did not trust the city of Salem and did not trust the information we were putting out. So reach out to your business chambers, your religious leaders, your neighborhood association liaisons, whoever you have that are trusted pieces of your community that folks will go to get accurate and current information. Use your employees. Use them, communicate with them early and often. As an employee, they're getting a lot of questions from neighbors, friends, families that are saying, hey, you work for the city, what's going on? Tell us what's happening. What do you know? So use them as ambassadors as well to spread good, accurate information or to tell people where to get that information. Another piece of really valuable information is to provide media training for staff if you intend on using your utility staff to do media, because often in the heat of the moment, if you've not been trained to do press conferences or respond to the media, it's a lot of pressure, and it for people who have not been trained in it, it can be really overwhelming. So identify early who that or those staff people will be. Get them training so whenever there is a communications event, they are ready to work with the public and provide adequate information. So the picture there that you see on top, that's myself and our emergency manager, and we were getting grilled by reporters at a press conference. It was a super uncomfortable event, and I'm very passionate about what I do, and it was a very, very difficult process to go through. And then the picture below was our Joint Information Center. So you can see a lot of people there actively working on all sorts of communication pieces for the city. And I think with that, I am actually at time. So this is just another beautiful picture of a bloom in our area. But I'm happy to take any questions that you might have.
Thank you so much. We do have one question in the chat that came in. It said, he says awful early in the year. Did you have a large increase in temperatures over a short period of time?
Yeah, so great question. We do start monitoring, actually, the city of Salem starts monitoring at the end of March, early April. And we typically do see a bloom, it's usually Dolichospermum, that happens in May. And so it's something it's pretty routine for us. It could be how the dams are operated. There's a big drawdown in the dams are there for flood control. And so during the winter months, they lower the reservoir significantly to be able to capture flood events. And so usually about April, May, they're back up to their top elevation. So I am wondering, if it has to do with that. We also tend to see our temperatures increase right around that time. We start to get some warm weather, which helps bring those surface temperatures up a little bit.
Thank you, Lacey. Anybody else have a question for Lacey? Please feel free to unmute yourself and ask a question.
Hi, this is Paul Suska. I do have a question. I noticed, well, first of all, I really like the different levels, you know, going from watch to the do not drink, all the different color-coded levels. I noticed you used algae, you know, the word algae rather than cyanobacteria, and wondering, I presume that was a conscious choice, and wondering why you used that term.
You know, I think for me, it was probably, I don't know that it was a conscious choice. Sometimes I use them interchangeably. You know, we, just like any water body, right, we're going to see a wide variety of algae and cyanobacteria. So we, you know, the data that we're getting in, you know, we are trying to focus in on the cyanobacteria, but we do capture all the other algae and you know, pay attention to that as well. You know, we've been impacted by the filter clogging algae, so there are other implications for us to be monitoring algae conditions up there. So yeah, sometimes I use the terms kind of interchangeably when maybe I shouldn't.
Hi, Liz. This is Leslie. I have a comment.
Yeah.
Hi, Lacey. This is Leslie D'Anglada with EPA. Thank you for sharing your experience with us. I still can remember that moment. I just want to clarify that you show a table that says EPA guidance values, and as I mentioned previously, we have only developed guidance values for microcystin and pseudomonas. And so we have not developed guidance values for cyanotoxin, cyanotoxin, or guidance values for dogs. So I'm assuming those are from Oregon or someone else. So I just want to clarify that only the guidance values at the bottom of that tables are the ones that EPA developed. And talking about guidance values, I know, trust me, I know how difficult conveying the message of having two different values for two different populations is. But I also want to say that EPA developed frequently asked questions, fact sheets, and other documents that are included in the risk communication web page that Katie mentioned, that explain what the health advisories are, and why we decided to develop two different guideline values for two different populations. And so I did not talk about health advisories during my presentation, but I'm just going to take 1 minute to explain a little bit about the health advisories here, just in case some questions comes after, and I need to leave the meeting in around 10 minutes. But so we base the health advisories on health effects, on the possible health effects that could happen based on the ingestion of water. So we know that infants, especially bottle-fed infants, ingest more water than adults. And that pattern of drinking more water than solid food goes all the way around 6 years old. That's why that we have one value for bottle-fed infants all the way to school children age, and then we have another one from 6-year-old to adults, because you start having more solid foods and you ingest other drinking that is not water when you're an adult. So your ingestion of drinking water per se is less based on your body weight. So the risk, the possible risk of having non-cancer effects from exposure to cyanotoxins is higher in people that ingest more water. That's why the values are lower for those particular populations. And we know that people that have kidney disease, liver disease, even pregnant women, and again, small children and people who are in dialysis are at a higher risk because they already have some conditions. So that's why we have two set of values. And so we thought that providing an additional value, right, for adults could also help in public health measures. For example, if your water system only feeds people in a prison or people in a community center or high schoolers, for example, there are a population that may not be at high risk for a lower value. So you don't need to issue a do not drink, etc. So you can take other public health measures depending on the population that is served. But again, we understand that the message could be confusing, and that's why when we provide that risk communication, it needs to be someone that knows about risk assessment, maybe a risk assessor, a toxicologist, someone that could really help the drinking water operator, the environmental health agency, the public health agency, and the media convey the correct message of what the health advisory means. Thank you.
Yeah, that's a great point. And I appreciate that. You know, we did partner with our county health departments as well as the Oregon Health Authority to try to distribute information. And I would encourage all of you to, the EPA has put out some really good communication facts and templates. So I encourage you to know where those are early, prepare early, have a plan in place, because in the midst of it happening, you know, it's better to have that in your back pocket and ready to go rather than to be scrambling to try to find it. I do see a question that I do want to answer very quickly because I think it's important for utility folks to hear. The question was, did you have ozone generators on site? We did not, but because of this advisory, the city has invested $80 million in on-site ozone at our treatment facility as part of our treatment process. And that ozone facility has just been completed. We're in the commissioning phase right now and will be up and online early next year. So we did make a significant investment in our treatment process following this because there were such huge ramifications from the public and their loss in confidence of drinking water that we still, I mean, it's been 3 years, we still have people who will buy bottled water. They will not drink our water. So trying to rebuild that confidence is huge, but also making the additional investment in ozone to help build that confidence back up.
Thank you, Lacey. There's one more question I think we can fit in, and then if everybody doesn't mind, we'll just take an abbreviated break because we are running over. But the question is, did the pack work or did the cyanotoxin concentrations from the source change?
So this is very interesting. What we think happened, so we did institute the pack. Our treatment facility is not designed for pack, right? So we were trying to put a treatment process in place that a facility wasn't designed for. We use a filter as a roughing filter, so a pre-filter filter, if you will. And when we started to add the pack to the treatment process, it clogged our roughing filter significantly. And so there were some implications with that, but what we found, we started to add acetic acid to the filters, which is a carbon source for the biological layer, that Schmutz deck. And what we found was that even with the addition of that acetic acid onto the filters, we were able to build back a really robust Schmutz deck, and it was able to take care of everything. And so we actually didn't need to utilize that pack after all our slow sand filters ended up doing their job and taking care of those cyanotoxins. But of course, still made us leery, which is why we've added in the ozone process.
Thank you, Lacey. Okay, everyone, I know we're running a little bit late. I'm just asking if you don't mind if we can do an abbreviated break, maybe come back in about 5 minutes, 5 to 7 minutes. That would be great, so we can continue on with our next speaker, who is Tom Waters. So we'll see you in about 5 minutes, if that's okay.
Liz, are you still there?
Leslie or Lacey?
Sorry, Liz. Hold
Oh,
on.
Liz.
Oh, hi.
Hi, this is Pierce. Hi, Liz. Are you there?
Hi. Yep. Can you hear
Okay,
me?
yes. Yeah. Are you all set with the discussion piece? I think you had some things about the questions, the poll questions.
Yeah, did
Yep.
you see what I had sent you
I saw you saying
in the
there
chat?
was an issue. Yep. I'm not sure I fully understood it, but you can show the discussion points after each question. Okay.
I'm
The
gonna
audience-
do I'm gonna do all the poll questions for the section and
Mhm.
then go into the discussions after that. Because before I was gonna do poll question discussion, poll question discussion. I'm just gonna do all the poll questions at once and then go into the discussions of each.
Okay, so instead of doing a discussion after each question, okay, is it because it's easier, or is there a problem? Why are we changing it? I'm just curious. Doesn't really matter.
Because I realized during this that if I do poll question discussion, the audience is gonna. Oh, Pierce, let me.
Yeah, that's fine. I'm just curious. We can chat later. But thanks.
Okay. Yeah.
It's going well. Thank you.
Okay, great. Thanks.
Hey, Liz, this is Lacey.
Yes.
You need me to stay on for the remainder of the workshop?
If you want to, you can. If you don't have time, that's fine.
I'm happy to do it either way. I just didn't know if you thought that it would be valuable for me to stay on or not.
Well, I think it would be if like when we go through the scenarios, if they have any questions about certain aspects and where you've been through
Yeah.
kind of what we're going through. So if you could, that would be wonderful. But I understand if you're busy, that's fine.
I'll plan to stay on.
Oh, great. Thank you so much.
Yeah, no problem.
Hi, Tom. Excuse me. Hi, Tom. Are you there? Tom? Tom Waters? If you can hear me, you're self-muted, so I can't hear you. I don't know if you're back from break yet.
Hey, I'm here. This is Tom.
Oh, hi, Tom. Okay, I'm going to turn controls over to you.
Okay.
And you can put up your PowerPoint and I will introduce you. So our next
Okay.
presenter. Oh,
are you seeing my screen now?
I am seeing your screen. Yes.
Okay.
Yep. You still are. Yep.
Perfect.
Perfect.
Sorry.
Yep. Great. Thank you.
Okay.
So, excuse me. So our next presenter is Tom Waters from the US EPA. Tom Waters is an environmental engineer with the US EPA Technical Support Center in Cincinnati, Ohio, with over 13 years of experience in drinking water. He works with the EPA's area-wide optimization program, providing technical assistance to states and water systems on treatment and distribution system optimization, with particular focus on cyanotoxins in drinking water. He has a Master of Science in Environmental Engineering from Lehigh University and is a professional engineer in the state of Ohio. Whenever you're ready, Tom.
All right. Thanks everyone for having me. I have a lot of material here and I know we're running behind schedule, so I'll try to go through this quickly. But if anyone has any questions, either afterward or I have my contact information at the end, feel free to reach out separately. Just a standard disclaimer for EPA, I won't go through that. Anything I say is just my opinion. So this is a summary of what I hope to cover during this talk. I'm going to go through just kind of an overview of treatment for cyanotoxins, and I'm going to try to cover several different types of technology. Most of my presentations focus on conventional treatment, coagulation, sedimentation, and filtration. And I'll talk about how both cyanobacteria cells and toxins can be removed through those processes. Also, looking at how ways toxins can propagate through a treatment plant and ways that you can sample throughout a treatment plant to evaluate performance of the treatment. And then I'll get into more focused on toxin removal by adsorption, by powdered activated carbon, and oxidation. And I will, and that includes chlorine, free chlorine. And then the final bullet there, I have some extra slides on some more non-conventional treatment processes, like membranes, slow sand, things like that, just to try to cover the range of different types of treatment technology that are out there. So I'll start with conventional treatment, and by that I mean I'm not sure if folks can see my cursor or not, but flocculation, sedimentation, and filtration. So you'll see that figure a couple times throughout my presentation. I'll just kind of show where in this sort of oversimplified treatment plan I'm talking about. Just a couple definitions that I'll mention just so folks are aware. Cell counts is just counting cells directly under a microscope. Chlorophyll and phycocyanin have been mentioned previously by previous presenters. Chlorophyll is a pigment that's in algae, cyanobacteria, and it plays a role in photosynthesis. Phycocyanin is unique to cyanobacteria, and that's why it's a helpful pigment to analyze for cyanobacteria. And then microcystin. And I'll talk a little bit about other cyanotoxins too, but this is probably stuff that folks know already. When I talk about intracellular toxins, I'm talking about toxins that are contained within the cell, the cyanobacteria cell, still within the cell membrane. Extracellular toxins are those that have been released from the cell and are in solution in the water matrix outside the cell. And then combined is just the sum of extracellular and intracellular. So just a couple terms that I'll be going through. And then, so I'm going to start with the conventional surface water treatment process. So again, by that I mean flocculation, sedimentation, and filtration. So just a couple introductory slides just to show how this set of processes perform at removing. Here, the point of this slide, and this is taken from a study by, I have it cited down here, but this is just to demonstrate how effective the conventional treatment process can be at removing cyanobacteria and thus, if they're intracellular, at cyanotoxins. So you can see, 2 different trials conducted with microcystin LR. You can see, the combined toxins were reduced through the treatment plant from 119 micrograms per liter down to 3, in the second trial from 60 down to 2. However, as far as the extracellular portion, you can see there's, no removal there. And so the message here is that, coagulation, sedimentation, and filtration are very effective at removing particles, cyanobacteria cells, in this case. So when toxins are located intracellularly, this process can be very effective at removing both the cyanobacteria cells and the toxins contained within the cells. Along the same theme of just sort of demonstrating the efficacy of conventional treatment, this was from a study by a colleague, Nick Dugan, at EPA, and he conducted a sampling effort at several water treatment plants that use Lake Erie as a source, and he used chlorophyll A as an indicator of cyanobacteria biomass, and sampled throughout the bloom season at all of these treatment plants. And this is just taken from one of the treatment plants. And again, the message here is that, most of the cell removal work, as indicated by chlorophyll A, is accomplished prior to filtration. So through the sedimentation process. So again, it's an effective process for removing cyanobacteria cells and the intracellular cyanotoxins. So one way to evaluate coagulation, how can you optimize that for cyanobacteria? Jar testing is a very effective tool for that. And to evaluate your optimal coagulant and polymer for cyanobacteria cell removal, these parameters I have listed here have been shown in the literature to predict coagulation performance pretty well. So in addition to turbidity, you can look at organics, pigments, color, UV254, which is an indicator for organics, particle counts, and streaming current or zeta potential. So I'm just going to highlight a quick case study that we conducted at a treatment plant in Ohio on a Lake Erie system. And this was a sort of a part of a larger study that we did with this plant on HABs. This jar testing case study we had 2 objectives. One was to understand the effect of the coagulant on cyanobacteria cell removal. Does it do as good a job at removing cyanobacteria cells as it does for other things like turbidity and organics and things like that? But also understand the effect of permanganate. This plant added permanganate as a preoxidant, and they claimed that they added it to improve the coagulation. So we wanted to test if that indeed was the case, but also test whether or not cyanobacteria cells were releasing toxins due to the permanganate addition. So we set up 4 jars. You can see them pictured here. And we stir them at mixing speeds equivalent to the turbulence in the raw water main. And so again, they're adding permanganate at the raw water pump station here. So we wanted to test, the conditions that the cells would see, representative of the conditions the cells would see in that raw water main to the plant. In order to test a sort of a worst case scenario, we concentrated a sample, a raw water sample using a phytoplankton net so that we had a higher concentration of cyanobacteria, kind of to test what the worst case could be for this plant. We added coagulant at the plant's dose for the day. We also added permanganate at the plant's dose, but also the high range of what they would add. And so I'll explain that in the next slide. So this slide shows both the results of the jar test, but also the experimental setup. So we have here, there's essentially 5 sets of bars. The first is just the concentrated raw water sample. We took this right out of the 5-gallon bucket, we mixed it in. And so the objective of testing, essentially jar 0, which is just our raw water sample, augmented with the concentrated cyanobacteria. And then in jar 1 was just the difference of stirring. And so there was some thought that maybe the stirring, the mechanical shearing from the paddles might induce a little bit of cell stress and toxin release. And so since we had it, we figured we would test that. But we also in jars 2, 3, and 4, this is where we varied the variables. So we have In jar 2, it's just the raw water with the plant's coagulant dose. It's aluminum chlorohydrate at 24 milligrams per liter. Jar 3 is the same conditions as jar 2, but also with the plant's permanganate dose for the day, which was 1.2 milligrams per liter. And then we also tested a higher dose just to get a feel for, if they do take the permanganate dose up to 3 milligrams per liter, which for them was the highest they would go. What effect does that have on the cyanobacteria toxin release? So you can see, again, the main objective I have of just showing this slide is just to show how effective the conventional treatment process, the coagulation process here, is at removing cyanobacteria cells. So you can see the difference between jar 1 and 2 is pretty big, as far as total microcystins. So that's the sum of both intracellular and extracellular. It decreases quite a bit. But you also see some extracellular here. If you look at between jars 3 and 4, you see some increases in the extracellular microcystin, which would indicate perhaps that the permanganate is, causing some cell stress and toxin release. Another way of, assessing your treatment performance during a HAB or prior to a HAB can be unit process sampling and what we call developing a plant profile. So in this slide, I'll talk a little bit about. Well, I'll just go through these. So the why we this was done at the same Ohio plant as I showed previously, and we had an exosond. I think Amanda mentioned a plant that used one of those earlier today, but this sonde was equipped with the sensors listed here. The two most important being chlorophyll A and phycocyanin, just because of their relevance to. Sorry, was there a question? Oh, okay. Just because of their relevance to cyanobacteria. And so we actually just dipped this sonde, let it equilibrate to the conditions in situ at the following locations in the plant. So we sampled in the raw water, the wet well of their raw water pump station, pre-sedimentation, clarifier effluent, and then combined filter effluent. And this can be done, this doesn't have to be done with a sonde, it can be done with grab samples, just any way that you can, take samples at each unit process to evaluate how well it's performing during a HAB can be very, it can be enlightening. So these are the results of that, just that using that sonde. I guess the things to pay attention to here are the different colors in the bars. I have chlorophyll listed as relative fluorescence units, but this sonde we were also able to calibrate to micrograms per liter of chlorophyll. We did not have, we weren't confident in our calibration for phycocyanin, so I'm just listing the relative fluorescence units here. But the main message is really just the differences between each unit process, not so much the magnitude. And then also we pulled samples grad samples for microcystins. And again, the message here, these just to clarify, these clarifiers are all in parallel. So it goes preset and then clarifiers are in parallel and then it goes to the top of the filter. But the big takeaway here is the difference between raw water and then what's coming out of the clarifiers. So large reduction in chlorophyll, large reduction in phycocyanin indicate it's the conventional treatment process is working at removing cyanobacteria cells and intracellular toxins. So at the end of each sort of technology, I'm going to list some operational considerations. So, understanding how effective a technology is at removing cyanobacteria cells and toxins it's helpful, but there are also some operational things that affect how that technology performs. And so I just want to highlight some of those. When a process is optimized, it's going to perform better, and so you can do that through jar testing, and optimal doses for daily treatment may change during a cyanobacteria bloom, and so it's important to do jar testing on a regular basis. Filters. I work with the area-wide optimization program at EPA, and we sort of emphasize a filtration goal of 0.1 NTU. So we've, and this has been proven in the literature as well, is that filters that regularly achieve an effluent turbidity lower than 0.1 NTU, they're going to be better suited when a cyanobacteria bloom does happen. They're going to be better suited to remove them. Obviously, these are, I think the next couple are probably pretty obvious. Backwashing filters is going to have to, the frequency may have to change during a HAB. So basing the filter runs on water quality data rather than just length of time of service can be helpful. And then more frequent clarifier sludge removal may be necessary during a HAB as well. I'm going to talk about adsorption, mainly powdered activated carbon, because it can be added. It can be retrofitted and be added into a plant if there are issues, unlike like a GAC filter. So just a quick overview on how PAC works and what affects its performance. It depends on the type of carbon, the source material, the type of cyanotoxin or other compounds to be absorbed, how much you add and how long it has to react, and then any interferences with competitive absorbents like NOM. And again, jar testing can be a good tool for assessing PAC type and dose as well. And AWWA has a tool, like a protocol as well as a spreadsheet for doing that, and we found that to be helpful as well in informing our PAC jar testing. And I just have this figure here to the right to show. Some of you may have heard, wood-based PAC for microcystins is found to be the most effective, but it really boils down to the pore size distribution within the PAC because of the microcystin LR molecule size, and so it not so much the source material, but its pore size distribution, because we're finding in lab studies that other types of carbon can also be effective as long as the mesopore size relative to the target toxin is they fit. So just a couple slides to demonstrate how the efficacy of PAC with cyanotoxins. Again, I'm focusing here on microcystin LR. So here we did this at a plant. We went to the plant, collected a raw water sample, and then spiked neat microcystin LR. This is something you can buy from a chemical vendor. And we tested various doses of PAC in this raw water, and then measured extracellular microcystin residual concentration. And this is pretty as expected. The more PAC you add, the more removal you get over time. I guess one thing to note here, well, two things to note. One is, you can see most of the removal at the higher doses happens at this plant with this type of water happens within the first 2 hours, and so that's something we would probably note for this plant. You don't need the contact time in this plant was 8 hours, and so we ran it out that long, but they probably don't need that much for this type of water. The other thing is that if you look at the 40 milligram per liter, we started at a concentration of about 63 micrograms per liter. And after 8 hours, it's down to about let's say 2-3 micrograms per liter. So that's still a high level to be going through. So even at 40 milligrams per liter, when you're doing this in raw surface water, a high dose of PAC still doesn't take you all the way down to zero. Obviously, it's not a magic bullet. And so there's many variables and also some operational considerations with PAC doses, especially at that high at 40 milligrams per liter. Here's another slide just showing the efficacy of PAC. The black dots, the filled dots are without PAC, and then these half dots are with PAC. So you can see extracellular toxins, when you add PAC, it does work. It absorbs. This is microcystin LR. So I mentioned some operational considerations for PAC, especially, I think Katie mentioned, just being prepared for harmful algal blooms. So consider how much you may need in the event of a HAB, how much storage space you have for the PAC, and any safety considerations. Do this all prior to HAB season so that you're not doing it, while there's a crisis. But also the operational impacts of adding PAC. I showed the slide of 40 milligrams per liter, and that's kind of the guidance that Ohio EPA uses. Have their systems. They recommend that their systems can have the ability to add up to 40 milligrams per liter of powdered activated carbon, but there's also some other impacts. You have to consider whether you'll be able to settle the pack out in your sedimentation processes. Will pack, carry over to filters? Will that reduce runtime? So several considerations. It's not just a matter of adding as much pack as you possibly can. So considering other treatment processes is important as well. I'll move on to pre-oxidation, mainly permanganate, because that's, as far as a pre-oxidant, we see that quite a bit. So these are just a couple studies showing the efficacy of permanganate and its effect on cyanobacteria cells. And so this is from my colleague Nick Dugan again. Here they dosed, in 3 different samples, increasing amounts of permanganate, and then he measured cells with damaged membranes. So he did that with a dye and measured or counted how many cells had damaged membranes. So you can see that as you increase permanganate dose, permanganate does affect cell integrity, which would then correlate likely to toxin release. This slide, it's a little confusing to show, but permanganate's also a very effective oxidant for extracellular microcystins. And so here you can see, as you add more and more permanganate, you're getting both with the low dose, you release quite a bit of toxin. So this is extracellular microcystins in a solution of cyanobacteria. Adding any permanganate causes quite a bit of extracellular toxin release, but then at the higher doses, you get degradation. And so that's something to consider as well. So just some operational considerations for permanganate. Due to what I showed previously, that large increase in extracellular microcystins due to the, even a low dose of 1 milligram per liter. Most of the time we tell systems to reduce or stop pre-oxidant to minimize toxin release, just for the duration of the bloom, so that they're not creating a bigger problem downstream in the plant. Consider, if you are ramping back on the permanganate, there's other treatment objectives that permanganate was likely achieving. So, how does that affect your turbidity, TOC, iron and manganese oxidation, any algae control within the plant? So just consider competing treatment objectives if you are planning to reduce the permanganate during a HAB. And so again, this kind of reemphasizes Katie's point earlier about just planning for and considering how these objectives will be achieved prior to the bloom is really critical. I'll move on to chlorine oxidation. So I like to show this slide. We get a lot of questions, especially when systems are having issues as to, my process, I use this oxidant and is it going to be effective? This is kind of like a literature review summary that was put together by Ohio AWWA and Ohio EPA. So I consult this a lot, actually. It's a pretty comprehensive table, but I'll just highlight, for microcystins, chlorine is effective, but it's as are many reaction kinetics for oxidants. It's pH dependent, temperature dependent, and then the presence of NOM can affect its efficacy just with competitive oxidation. One thing to note here too is just the ineffectiveness of chloramines and chlorine dioxide. As I mentioned before, potassium permanganate across the board, with the exception of saxitoxin, is pretty effective, especially for antitoxin and microcystins. Ozone, I have listed here for systems that use ozone, and then UV advanced oxidation. I have them combined because UV by itself has been shown not to be effective for oxidizing cyanotoxins. So there's a lot I want to talk about on this slide. This is from I have the citation down here. It's kind of the benchmark study for oxidation of microcystins with chlorine, and it's used in a model that AWWA has as well. It's called CyanoTox, which is very helpful for assessing CT for cyanotoxins. I'll mention that later. But here, I want to make a couple points. Hopefully, it's not too confusing to see. So the y-axis is CT, so that's concentration of oxidant times the contact time. That's necessary to reduce microcystin LR from a concentration of 10 micrograms per liter down to 1. And 1 was the target when this study was conducted. 1 was kind of it was the World Health Organization. It still is the World Health Organization, finished water target. So that's the significance of that. So you can see that this reaction, this oxidation reaction between chlorine and microcystin LR is both temperature dependent and significantly pH dependent. So at lower pHs, a much lower CT is needed for taking the microcystin LR concentration down from 10 to 1. However, if you're at pH 9, it's a lot higher, order of magnitude higher. The takeaway here, or I tried to highlight them here, is just you go from, if you increase from pH 7 to 8, there's a 2-time increase in CT, and then from pH 8 to 9, it's a 3-time increase. And that's significant because this slide was actually developed with Nick Dugan again. We thought it'd be helpful to put in sort of a reference here, CT for 3-log GRE inactivation at 1 milligram per liter of chlorine at a temperature of 25 degrees C for comparison. So at pH 7, for 3-log GRE inactivation, your CT is 37. So if you're at pH of 7, you're probably already adding chlorine, through your, for GRD inactivation is probably high enough already. But at higher pHs, a much higher CT is needed than you would be achieving for GRD inactivation. So it's really important to, during a HAB, consider this when you're if you're hoping to use chlorine as a way of reducing microcystins in the finished water. So a way to do that, well, I'll mention I'm going to skip forward to Cyanotox here. This is the model that AWWA has put together. It's really effective. You can put in your Cyanotoxin. They have microcystins, cylindrospermopsin, anatoxin, and saxitoxin all in there. You can input your pH, your temperature, initial concentration if you know it in your raw water, and then what your goal is in the finished water. And then what type of oxidant you have. I think it evaluates free chlorine, chloramines, permanganate, and I'm drawing a blank on the last one. I think it's ozone. But this is all essentially they pooled lots of literature studies on the kinetics of oxidation with these different variables and put this spreadsheet together. And it's pretty helpful for evaluating CT that's needed. So I'll just go back to this slide to talk about a kinetic study that we did at a plant. This study that I mentioned here was conducted in lab grade water with just microcystin LR spiked. So there's no other constituents in the water aside from the chlorine added and the microcystin LR that's added. One of the problems with these kinetic studies is that they're not conducted oftentimes in a raw water sample that has other things that compete for oxidation. So a lot of times when we go to plants, we would be thinking we needed to add some kind of safety factor as to what CT this plant would need, to account for the chlorine consuming other things like NOM, iron and manganese, stuff like that. But we didn't know what kind of safety factor to add. And so what we did was we conducted a sort of a bench scale kinetic study with the system's raw water. Again, this was kind of a theme. So this raw water wasn't experiencing a bloom. So we had a concentrated solution of cyanobacteria obtained from another water body that was experiencing a HAB. Freeze thawed the sample so that we would have, ensure that all the toxins were extracellular. And then we added chlorine at the plant's dose and measured microcystin, the free chlorine residual and microcystin in the water over time. And then compared it to the Cyanotox model. And essentially what we wanted to understand was the difference between, how this plant's raw water behaves versus just, like a lab study. And, the difference is really just what's going on with competitive oxidation. So the results of that are here. It's again, it's a busy plot, but if you just focus on the green, you know, this is microcystin, and then the dotted lines are what's predicted by the Cyanotox model. You can see over time. So we added the y-axis is chlorine residual over here, and then you can see microcystins. I have it on a 2-axis plot, and then time is on the x-axis. And so over time, microcystin degrades. These are the actual samples, down from about, I think we had just short of 4 micrograms per liter initially, down to about 0.25 micrograms per liter by 5 hours. However, based on the Cyanotox prediction, you know, the model, the kinetics predict that, you know, we should be at 0 microcystin LR, or this is total microcystin, sorry, total microcystins by about 3 hours. Based on, you know, just the reaction kinetics alone. And so the difference here really is, you know, the competitive oxidation. The raw water in the system likely had some other things in it that was also reacting with the chlorine. So just lots to think about there with chlorine, but you know, consider you know, where it's dosed and if any other competing technologies are present. You know, what are your organic levels at the point of chlorine dosing? You know, is there ammonia in the raw water? Do you have GAC filters downstream that, you know, if you're adding chlorine that could reduce its effectiveness? And then, you know, consider the potential for formation of DVP. So, you know, if you're increasing your chlorine dose, how's that going to affect your DVP compliance? So, again, you know, multiple objectives here, and it's hard. I'm seeing that it's almost lunchtime. I'm sorry that I'm going so long. Just a couple. I'm going to talk about filtration here. Non-conventional filtration processes. I only have like 5 more slides, I think. Slow sand. The main message here, I can make these slides available to folks afterward, but the main message here is that slow sand has been proven to be effective for both removing cyanobacteria cells and some portion of the extracellular toxins, mainly due to 2 processes. One is the physical filtration of the cells and intracellular toxins, and then biodegradation of the extracellular toxins. And there's several variables there, you know, to consider, but that's just the main message of this. Membranes, the main thing I want to convey here is that, you know, low pressure membranes, you know, micro and ultrafiltration membranes can remove the cells, but the pore sizes are too big to remove the toxins. However, high pressure membranes, nano and nanofiltration and reverse osmosis are capable of removing extracellular toxins by you know, a combination of size exclusion and charge effects. I'll reference the area-wide optimization program again. We have goals, you know, goals for turbidity removal from membrane filtration plants of 0.05 NTU, and so again, it's a good benchmark to alert operators. You know, if to operate, it can indicate issues in between direct integrity tests, and so having a goal, you know, like a goal for your baseline operation is helpful. You know, when it comes to treating for cyanobacteria and cyanotoxins. I'll just talk about the clear well here real quick. Just not sure why that's, I think these slides are out of order, I apologize. UV, we get asked questions, especially recently about, you know, is UV effective for cyanotoxins? And again, I'm talking about the toxins here. UV has been shown to be effective for inactivating cyanobacteria cells, but as far as toxins go, and usually where the UV is applied in the treatment plant, it's hopefully by that point it's mostly extracellular, you know, dissolved cyanotoxins. And so the literature has been pretty conclusive that that while UV can degrade cyanotoxins, the dose that's required is so high that it's really not practical for you know, for water treatment plants. And you know, again, we can talk offline if folks are interested more about that. But chlorine dioxide, I showed on the table previously. It's a very slow reaction with microcystins, especially, and then ozone. It's a very effective oxidant at degrading microcystins, as well as cylindrospermopsin and anatoxin. So I just have some conclusions here. I know that we're kind of behind schedule, so I'll just show them, and then if folks have questions, I can address them. I'm not sure if I'll have time here, or feel free to reach out. And so here's my contact info. Appreciate the time. That's all I have.
Hi, Tom. Thank you. Excuse me. So we are going to move the first scenario to after lunch because it is relatively short. So we'll have time to do all three after lunch. So Tom, we can take some questions because we'll break for lunch when people are done asking. We do have lunch scheduled from 12 to 12:30. So we should have plenty of time after lunch to complete, the exercise with all three scenarios. So if anybody has some questions for Tom, please feel free to unmute and ask your question. I know that Ian from Rochester put a question in the chat kind of at the beginning of your presentation, Tom. And when you were talking about the study, he wanted to know do you know what the target pH was for coagulation in that study?
I don't. I assume, I'm just trying to get back to that slide. I wasn't sure what slide it was, but I'm not sure what the target pH was. I can, hold on. Yeah, I showed a couple. I'm not sure. These are literature studies. I'd have to go back and consult them. This was not sure what the pH here was. And then this was a jar test study. And again, I'm not sure what the target pH was. But yes, I know that is a variable to consider. These were a lot of, some of the studies I showed that we conducted, we conduct them as part of larger, like comprehensive evaluations of treatment plants. And so a lot of these studies happen very quickly in order to get just some high level sort of ballpark results, which we then use to kind of direct further study for the utility. And so a lot of times we are missing information.
Thank you, Tom. Does anybody else have any more questions for Tom? Please feel free to ask your question.
Hi, this is Paul Suska. Tom, when jar testing, what's a useful parameter to use for performance? Can you use something that's less expensive than actually testing for toxins?
Yeah, that's a great question. I tried to cover that a little bit here on this slide. Hopefully folks can see that. And there have been some studies out there. I know there was one in 2015 by Gail Newcomb, where they actually had like a formula for, I think it was a combination of NOM, color, and UV254. But yeah, so you can look at turbidity, you can look at organics like UV254. Sometimes those kind of correlate with if you think about like what may make up like total organic carbon. Some of it could be intracellular matter that comes out. But I think we've had probably the most success with the pigments chlorophyll A and phycocyanin. You can test those with like a benchtop fluorometer, pretty quickly as well. So there's there wouldn't be a delay as far as testing for toxins. Again, the chlorophyll A can be an indicator of total photosynthetic biomass that's in the water, whereas phycocyanin is produced only by the cyanobacteria, and so that would give you a better idea of what's going on with cyanobacteria biomass. And we've thought a little bit about how you process the samples, whether if you if you do like an in vivo versus an extractive
Hi again, everyone. I hope you had a good lunch. So now for the rest of the event, we're going to go into the scenarios, and what will happen is we'll show a scenario, then we'll have some poll questions that are relative to that scenario, and then we're going to open it up for discussion. We'd like everybody to be involved. If you have a question, suggestion, comment, anything, please feel free to just unmute yourself and join in. So here are the guidelines. First of all, try not to fight the scenario. You want to treat the scenario as if it could affect your source. Again, we'd like to have you actively participate. Think about your current plans and policies and procedures that you may have in place for such an event or an emergency event. Apply common sense. Respond to the questions, based on your knowledge. Use your experience and understanding for the basis of the discussion. And again, just join in the discussion, share ideas, ask questions. This is, I just want to check, can everybody hear me?
I can hear you, Liz.
Yes.
Okay, great, thank you. Okay, I was just getting a message that somebody couldn't hear me, so I just wanted to check on that. Again, this is supposed to, meant to be a very casual, laid back information sharing event. So again, please feel free to join in. And after the event, you'll be getting a follow-up email with an evaluation of the event. So if you could please just take a couple of minutes to fill that out when you get it, we would appreciate it. And I also want to mention that Mike Unger, one of our engineers here at DES, is also at the event, and he'll also be available for questions and comments further on when we get into the treatment portion. So for the background of the scenarios, and it's, obviously hypothetical scenarios. So New Hampshire is now experiencing a warmer than average spring after the winter. Excuse me, my toolbar is blocking my view here, sorry. With a below average snowfall. Loon Lake supplies raw water to White Mountain Waterworks in 3 communities, Smithville, Jonesville, and Bear Village. A cyanobacteria bloom has been seen forming in Loon Lake about a quarter mile away from the intake. The wind conditions are moving the bloom closer to the intake, and White Mountain Waterworks uses conventional treatment. So that was the background. So for our first scenario, on August 10th, visual checks on Loon Lake indicate there's a bloom forming. On August 14th, the visual checks show that a large bloom in Loon Lake a quarter mile from the intake. And at this point, customers are complaining of taste and odor issues. And again, the wind conditions are moving the bloom closer to the intake. So the highlight for this scenario number 1 is there's a large bloom in the drinking water source. So now I'm going to administer some poll questions, if you could answer them, and then we will have a discussion about them after. So the first poll is, thinking about the New Hampshire DES protocol, who should be contacted to prepare for a possible cyanotoxins event? So if you could just make a choice. And also, if you have another idea, then please feel free to put your response in the chat. And I'm just waiting for some more responses. Okay. So I will now share. Okay, so about split, half say us here at NHDES, and the other half say about all of the above. Okay. So the next question is, do you know of an accredited lab that analyzes cyanotoxins using approved EPA methods? And this is pretty simple, yes or no. Okay. Great. So just about everybody knows of a lab that can do that, so that's great. And the last question for this section: what types of water quality monitoring should be conducted at this point? Okay, I'm just waiting for a few more responses. Okay. So we have answers 1, 2, and 3 only. We have a couple for daily. All right, great. So now we're going to go into the discussion portion. And like I said, if you have questions or comments, please feel free to unmute yourself and join in. So for the first one, when you're thinking about our protocol, who should be contacted at this time for scenario 1 to prepare for an event? And what should happen is you would contact Amanda at the JCLC. Amanda will contact us at the Drinking Water and Groundwater Bureau, either Paul, Pierce, or myself. And if in that time you're unclear, you can always reference to the CyanoHABs response protocol if need be. And then, do you know of an accredited lab that analyzes cyanotoxins using the EPA methods? Just about all of you said yes, and that's great. The two that are in our response protocol, Rhode Island Public Health Lab and Green Water Lab, are great to use. They can usually give a very quick turnaround time, which is necessary at this point to give us the analytical results that we're looking for. And then what types of water quality monitoring should be conducted? You definitely want to do daily visual surveillance. You want to check for changes in your water for color, turbidity, especially at the intake area. And if you're able to, you want to track changes in the pigment, the chlorophyll A and phycocyanin if possible. And you definitely want to take samples at the raw water at the bloom, the raw water away from a bloom, the raw water near or at the intake if possible, and then you'll bring those to Amanda at JCLC for cell counts and identification. And due to the taste and odor issues that you've been receiving complaints about, you would want to take a raw water sample and have that also analyzed for cyanotoxins. And again, we're stressing this is why in preparation of the Cyanobacteria bloom season, which is usually late spring and can go into late fall, you really want to be prepared for such an event that could happen. So visual surveillance should definitely take place on a regular basis, either weekly or biweekly, depending how vulnerable your source water is for cyano blooms. Now here's a slide that, because we're telling you to do visual surveillance. So exactly what is it that you're looking for? So this is a great source. This is from the guidance out of Vermont. So over on the left here, you'll see where they're doing what Vermont calls category 1. There's very little or no cyanobacteria present in the clear water. So you're just looking for changes in that turbidity, maybe changes in color. And as you see here, as things progress and the bloom gets more intense, the water's going to become more colored, more turbid, till you get all the way over here on the right, where there's a very high account of cyanobacteria present in the water. So when you are doing surveillance, this is the main thing that you're looking for, is basically color change in the water and changes in turbidity. Does anybody have any questions or comments at this point?
Hey, Liz, this is Paul. So it was interesting to note that, in the first question, so many people, their response would be to notify, not just the DES, but also the health officer and the public. And I think that we certainly wouldn't fault anybody for doing that, especially notifying the health officer. But it might be just worth noting that, given our experience so far in New Hampshire, it's unlikely that there'd be a health hazard. It's possible, and that's why we're doing all this testing and response, but it's not likely there'd be a health hazard, so it might be, getting the public concerned unnecessarily if the public were to be notified at this stage. So anyway, I just try to circle back to that and see if some of those maybe who responded that, that the health officer and the public ought to be notified, that it'd be interesting to discuss that point maybe.
Yes, for those of you that did check that option, would you like to discuss that at this point? I'm not getting any response.
Okay.
But yes, as Paul said, I know like different states have different guidelines for what they would do on all different types of scenarios. But as Paul said, in our experience here, what would happen if you went out and saw a bloom in your source water as presented in the scenario? At this point in time, you would just need to contact Amanda at JCLC. She would then contact us right away. Get the, at this point we're just gathering information, finding out what it is, what type of cyanobacteria it is, what the cell counts are, and in that point, then as you'll see in the further scenarios, that then we'll go from there as to what needs to be done next. And Amanda, do you have anything that you'd like to add to that?
Sure, thank you. Yeah, I think, everyone would be notified. It's just sometimes we'll get a report. It's not a cyanobacteria bloom. So we don't want to bother the health officer at that time. So what this program will do is try to get out there as soon as possible just to evaluate the situation. But, I'm basically calling Liz just as I hear about it. So it's all kind of happening at once. It's just sometimes it's a false alarm.
Great, thank you, Amanda.
So how, if you did get the call, how long do you think it would take for you to go out there? I mean, so how rapid is the response?
Amanda, did you want to?
Yeah, sorry, I was talking with mute. Thank you. Yeah, so we do have this hotline, which is pretty quick, is sometimes slower on the weekends, but I have a cell phone on me all the time to at least be able to reply and respond. We encourage taking a photograph just to kind of rule out if it's something, as a non-issue. But we are usually out there the same day or the next day. Sometimes, though, we do have multiple blooms all in one day, and so we're very limited and we would ask for help. So if it was a public water system, I would reach out to Liz and ask for sampling assistance. or might even ask the operator to bring in a sample, or we'd coordinate it some way. But, you know, it's been within 24 hours.
Does anybody else have any questions or comments?
Liz, we did get a comment in the chat from John of Canaan to identify it first and then act.
Right, and that's kind of what we're suggesting at this point, and what Amanda was saying is, let us get out there, get the sample, find out what it is, and then at that point, we can take it from there and find out what the next steps would be. Okay, no more questions or comments? Okay. So moving on to the second part of the scenario. So now we have the cyanotoxins breakthrough in finished drinking water for the vulnerable populations. And those include nursing homes, hospitals, daycares, infants, as Leslie and Katie had mentioned earlier. So on the 17th, Amanda at the JCLC has confirmed that microcystis is now at 100,000 cells per mil. The bloom is now on the 20th confirmed at the intake. Raw water samples have been sent to JCLC for analysis. A finished water sample has been sent to a lab for EPA method toxin analysis for finished drinking water. Then on August 21st, Amanda confirms that microcystis is now at 200 cells per mil, and another second finished water sample has been sent to a lab for EPA methods for finished drinking water analysis. On August 22nd, the first finished water sample results show microcystin toxin concentrations at 0.5 micrograms per liter, which is above the EPA's health advisory level for vulnerable populations, which is 0.3 micrograms per liter. On August 23rd, White Mountain second finished drinking water sample comes back and it shows microcystin concentrations also at 0.5 micrograms per liter. So the highlight in scenario number 2 is now we have a breakthrough in finished water. It's above the vulnerable population drinking water health advisory of 0.3, but it's still below the school-age children and adult advisory level of 1.6 micrograms per liter. So now I will post some questions regarding this scenario. So given this scenario, when should you start communicating about the event to the public? I'm just waiting for a few more votes. Okay. And I will share the results. Right, so just everybody says when you have a finished water detect at or above the HA. Okay. And the next question. So given there is 0.5 micrograms per liter toxin levels in your finished water, what actions should you take to manage the event? I'm just waiting for a few more votes. Okay. And everybody said all of the above. So communicate with public, communicate with local health officials, issue do not drink order for vulnerable populations, and modify treatment to potentially mitigate the toxins. All right. What treatment modifications would you consider at this point? All right. Okay, so we have distribution over a few of them. Okay, great. And the last question for this section. What are the finished water sampling intervals EPA recommends to know whether the toxin levels are increasing or decreasing? Would you want to sample weekly or daily? I'm just waiting for a couple more votes. Okay, so just about everybody said daily. Okay, so now we'll go into the discussion. So the first question, given the scenario, when should you start communicating about the event to the public? Yeah, so as soon as you have a detect of 0.3 micrograms per liter, you should notify the public and explain the health risks, especially to vulnerable populations. A list of vulnerable populations should be in your system's emergency response plan. So what this means is, as soon as you get that first detect of at or above the health advisory for vulnerable populations, you immediately want to notify the public and explain the health risks. And this is done because of a few reasons. For one, we may not know exactly how long the toxin has been getting through the finished water. So I know that there's been a lot of talk about that 10-day advisory. And that's really just a guide. It was studies that were done by EPA that they think for 10 days, if a vulnerable population was drinking the water and it was at 0.3 micrograms per liter, that they may not get sick. But because there could be lapse in times of multiple testings, and again, maybe toxins were getting through and for some reason we just didn't know, it's really critical that the vulnerable population be notified at that point. So it's as soon as you get that first detect of 0.3 micrograms per liter. Excuse me. So now, given there is 0.5 micrograms per liter toxins in your finished water, what actions should you take to manage the event? So because now, again, it is above the 0.3, and we know that it is now above the 0.3, but it's, it's even though it's still below the 1.6, for the general population, you would definitely want to communicate to the public and local health officials. definitely want to issue the do not drink order to the vulnerable populations. And you want to explain to the public everything you know about the bloom, the genus, the type of toxin it produces, any health risks involved, analytical findings, etc. You want to be as transparent as possible, and especially going back to what Lacey said from Salem, Oregon, they had not in through no fault of their own, but the public can lose trust in their public water system very quickly. So if you're being transparent, you're giving them all the information you have, telling them everything you know, that helps to maintain that trust between the public and your water system. You definitely want to advise customers to do not boil the water. Boiling does not affect these toxins like it does for other bacteria, say for E. coli. And boiling can break the cells and release these toxins even more into the water. So that's a very important thing, because I know a lot of people that I talk to, you know, they hear of all they hear is bacteria, and what they have been known in the past, oh, it's bacteria, I need to boil the water, and then it's safe to drink. So that's a very important point that you want to get out into the public. And as Katie had shown earlier, the EPA has a lot of templates and flyers and things that you can use to get this information out to the public, all these different templates. And they have templates for every different situation you could think of for what is happening with cyanobacteria in your source water. So that's a great place to go and have these templates on hand in case you need to make a public notification. And again, you definitely want to explain to the public what is being done to address the situation. And yes, you would definitely want to start to modify treatment to potentially mitigate the toxins. I know that Katie had put this up at one point, and here is a great flyer from their risk communication toolbox. It's very simple yet very powerful to let people know what is happening at what stage, and to let them know about what actions are being done. So I just wanted to put this up there to say this is an example of something you can get from the EPA's risk communication toolbox. And again, they have so much good information in there that can help out public water systems in a case of this. So now to get into the treatment modifications you would consider, and I'm going to ask Mike Unger to step in here to go over this portion of it. Mike?
All right, can you hear me, Liz?
Yes.
Great. So first, as Tom talked about, optimizing treatment for essentially particle removal. So for the systems that have conventional treatment, optimizing coagulation, and then or adding powdered activated carbon if the system's designed for that, and jar testing. There were some great examples that Tom showed of that to try to, again, we know that the conventional treatment process is effective at removing the bacteria themselves. And same thing for filtration. So if you're able to reduce the flow rate to improve the effectiveness of filtration, and then because of the loading on the filters, you may need to increase the backwash interval. I saw in the poll some people had said add an oxidant, and that it may have been thinking post filter. So that's the distinction here. As Tom said, the oxidant can damage the cell membranes and potentially release the toxins into the water. So the key is to try to optimize the coagulation and filtration, remove as many of the cells as possible, and then depending on what the toxin is, we did see that chlorine could be effective, or ozone. And then here's a excerpt from EPA's guidance to really look at each unit process and understanding the objective. So trying to remove the cells early in the process, and then if you're able to remove toxins through oxidation later, and some of the key things that Tom had recommended monitoring for the pigments are a good, I guess, surrogate might be the right word for the cyanobacteria themselves. Some of the systems that don't have a whole lot of sophisticated capability, there are some ways to monitor those processes and know that you're optimizing treatment.
Great, Mike. Thank you very much. And for what are the finished water sampling intervals recommended by EPA to know whether your toxin levels are increasing or decreasing at this point? Basically, at this point, we would be wanting to sample the finished water daily, because you really want to know if your values are decreasing or increasing. So it's very important to do a daily sampling of the finished water. Does anybody at this point have any questions, want to join in discussion? So we do have one question in the chat that says, don't we need to have DES approval to modify our treatment process? And Mike, I was wondering, could you possibly address that?
Yeah, so that's a good question. And in general, yes. If we're just talking about optimizing coagulation, then no, we would just consider that an operational action. Jar testing and optimizing coagulant dose or filtration rate. If it's a new process, like if you were to add PAC to a plant that doesn't otherwise, then yes. But I'm assuming at this point DES would be heavily involved. So we would be in discussions already, and we can approve a temporary change and in an emergency basis.
Great. Thank you, Mike. I see somebody. Anybody else have any questions?
This is Lacey, and I have some additional comments.
Sure.
Just to throw in, because this is what we went through.
Absolutely.
A few things to think about for your utility specifically that you can actually kind of do decision points now. These are vulnerable populations, so a lot of times this is going to affect, obviously, preschool age children, could affect elementary school, elderly care facilities, things of that nature. So, would you, as a utility, plan to do any sort of water distribution? So the minute that you send this message out, there's going to be a run on bottled water in your community, likely. And so, working to get in additional bottled water capacity to your city or location. But also, as a utility, are you planning to distribute water to any specific population, and how do you intend to do so? So just something to consider, because those decisions can be made now. And then also, as soon as you think that an event is happening, my recommendation is to document, because the media is going to want to know what you knew when and what you did. They're going to want a timeline of events. And that's going to be, it's better to take that information down as it's happening rather than to try to go back and create that timeline after the fact. So, who's notified when, what treatment processes were modified when you sent lab results out, when you expect those back. All of those things need to be really documented well, because it's likely that the media will be asking you for that information.
Thank you, Lacey.
This is Paul. Sort of taking off on Lacey's comment, which was really interesting about when, to notify the public, and having been through that experience, I'd be interested to hear what Lacey thinks about, I mean, I saw some headlines about, well, why didn't, officials notify the public sooner? What did they know and when did they know it, kind of thing.
Yeah.
Are you basically, damned if you do, damned if you don't? Or
Yes.
is there based on your experience, what do you recommend?
So everything in drinking water is very time sensitive, right? So the things that we were nailed on were the lab delay, right? So you have a bloom, by the time you get somebody out and you sample, you get those results back, and there's a problem. Okay, well, if that's 2 days ago and you sampled at your intake, has that water already made it through your treatment plant? Is it already in your distribution system? So then you're sampling at your finished water sample site, waiting 2 or 3 days to get that information back, possibly. And now all of a sudden it could have been in your system for, 4 to 6 days, and nobody knows anything about it. So it's very important to make an internal decision on what is your notification piece going to be. So we decided, we waited for confirmation samples, right? We got that initial hit. We wanted confirmation samples to ensure that this was indeed a real and ongoing issue. That happened to fall, right, over a weekend. There were shipping delays, all sorts of things, which extended that timeline. So actually, in response to this event, we also purchased our own ELISA machine and now have our own internal lab that's certified to run for microcystin and cylindrospermopsin. So we can actually get same day results because we were hammered so hard on that. As soon as you and your regulatory agencies are confident that this indeed is an issue, then you want to get your story out there fast. And one of the things that happened to us too, which we were trying to be good partners, it was the school year. We had notified the school district that this might be coming because we wanted them to prep to be able to get, figure out how they were going to do school lunches if they needed water, how are they going to, get bottled water to students, etc. And they took that information and they sent it out without us actually issuing an advisory yet. And so we lost control of our own story because we were trying to do the right thing and get that information out to a critical partner. And they sent it out to their employees, and then all of a sudden it just spread like wildfire. So we started out kind of behind the gate already. So as soon as you're confident there's an issue, as soon as you know that there's going to be something happening, you want to be able to issue that advisory as soon as you possibly can. So the things that you can do in between is get your press release ready, get your talking points ready, pinpoint who your media person is going to be. Do you have people who are trained and ready to go on social media to get information out? Who are your critical customers? You can get all that lined up and have that all ready to go. So as soon as we know, yep, this is an issue, you can roll. And then that also helps in your timeline, right? Because it's not like you're just sitting idly waiting for a test result in. There's all this work that's been going on behind the scenes to get you prepped for the event that you do have to go into an advisory. So that's why the timeline's real critical. When did you get your result? When did you let us know? When did you pull the confirmation? How come it took 3 days? Wasn't there a closer lab? Couldn't you rush the sample? There was boom, 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 all sorts of questions that, you can kind of preemptively know and make sure that your timeline's in place, because the public and the media will want to know.
So with 2020 hindsight, at what point do you go public? When you have raw water above the health advisory level, or sooner?
So City of Salem is because of the situation we specifically went through. We actually we put our raw water information on our website. So people know when we're sampling our intake, and they know what the result is at the intake. And then we also keep people up to date with that kind of red light to green light graphic, so they kind of know, like, oh, there is a bloom, they're paying attention, they've started this kind of treatment, you know, they kind of know where we're at in the spectrum of things. But by having our own on-site equipment to do the analysis, we can pull a sample that morning and get a result late evening. So it helps turn things around much faster for us to do the decision making and get that information out to the public. But we have that capability. We have an in-house lab that is now certified and can do that, turn that around very quickly. I realize that most utilities probably don't have that, that capability. So we turn things around very fast, and they get our raw water data as well. And I know that Toledo, Ohio also puts their raw water data online after their event, just because of trying to rebuild that consumer confidence. And West Palm Beach, Florida actually just went through an event this summer. So they're also, we're very much struggling with trying to get the consumer confidence built back up, and the lab delay, I think, was also an issue for them. You know, people, you know, people get upset. It's like I have a pregnant wife who's 8 months pregnant, and you've known about this. You got your first test result on Monday, and now it's Wednesday, and now you're telling us, and my wife has been drinking the water for the last 2 days. You know, so if she gets sick or something happens to our unborn child, you know, what are you going to do about it? You know, it's those types of very emotional, fuel-filled questions that the media really, you know, will latch onto and highlight, you know, kind of all the rationale, you know, instead of really, when you have a vulnerable population, it's a smaller piece of your population that's affected. For the majority of the people, the water's safe to drink. But that's not the messaging that gets portrayed right through the media. It's that all these people can't drink the water because there's something wrong with it. So your messaging is really, really important on how you deliver.
Great,
Liz, this is
Lacey.
Mike. Sorry,
Oh, go
I
ahead.
had a question along those lines. For the labs that you mentioned, what's the cost for the toxin analysis, and what is the turnaround time?
Right, so for Rhode Island Public Health, I have talked to them, and where we don't have, where they wouldn't be sampling like a bunch of samples, you know, every other week, basically, from us for all different sources. If they only get a few, they will do it for no cost. Their turnaround time is probably 2 days. We just have to notify them in advance that samples are coming. Green Water Labs, I do have the in our response protocol is the pricing for both of them, in case Rhode Island cannot do it at no cost any further. I believe for just one of the EPA methods that just does the ELISA, I believe it's around $50. For the 2 other EPA methods, which are usually combined, I believe I want to say around 200. But Green Water Labs sometimes can turn around same day, or 1 day turnaround. So as soon as they get the sample. So those 2 labs are usually pretty quick in trying to get the turnaround time. And again, I apologize, I can't remember the pricing completely, but it is in our CyanoHABs response protocol, what the pricing is. Also, how in the response protocol, also how to take a sample, how you would need to take a sample for these labs. All the information is in there for everything you have to do there. The lab's contact information. So everything is in there to tell the systems how they would take a sample and ship the sample.
Great, thank you.
Sure. Katie, I know that you had your hand up. If you'd like to jump in. Katie Foreman of the US EPA.
Hi, thank you. Thanks, Liz. And I just want to thank Lacey for sharing all of the really pertinent lessons learned and information on communication that I feel like was really valuable for folks to hear from a system that went through it and knows exactly what they're up against. if, you know, Lacey mentioned West Palm Beach had an issue this year. We've had over 5 different major emergencies this year across the country. And I'll just mention from lessons learned on the communication there is that we know that, you know, monitoring delays happen. And, you know, and a lot of places don't have that monitoring infrastructure in place until after, you know, they have a known issue or they've had an emergency. So a lot of people are in this what do I do moment, you know, we're waiting for lab results. And I'll just mention that, especially this year, we've been dealing with systems that at that first sample in their raw water where it's detected, they're communicating with the public, not that their water is unsafe, but that there is an issue in the source water and they're managing it this way, you can still drink it. So they have been communicated before the event has hit finished water. And in one case, that's all that had to happen. And that is, you know, building trust in your community is that you've been transparent, it's happening, we'll keep you posted, we'll let you know what the results are. And then it turned out they didn't have to issue a do not drink. There was another system that ended up having to issue a do not drink. After just one sample, they decided to do that because it would take 3 days to turn around another sample. And they had one hit in raw water or in finished water and communicated directly with the public, not waiting for a confirmation sample, but letting the public know that there was a confirmation sample that was set out. We'll let you know, but out of an abundance of caution. So there's a little bit there on these scenarios are very dynamic, but the more you communicate and communicating sooner, even, you know, if you're in a system that has HABs in your source water, getting the public to understand what they are prior to, you know, if it becomes an issue in their drinking water is really important. So I just mentioned there are a lot of different ways to communicate with the public. It's not just the do not drink. So recommend integrating some of that. If you are a system that has, has a HAB season and, you know, actively has HABs in your source waters throughout the year, letting people know treatment's working. I mean, that's a great communication too, is that we haven't had breakthrough, letting you know, we're dealing with this issue by doing these things. And there are lots of different places across the country where those communication tactics have ended up, and those approaches have worked. And, it doesn't, you're not necessarily playing that blame game of when and how, if you're constantly communicating, understanding there is an level of effort there. But I've just mentioned that when you're developing, what are you going to do in your communication plans? Getting a sense for how long those samples take is really important to know when are you going to wait for another sample? So just getting those logistics, thinking those through before the emergency, I think is really helpful. And again, thank you, Lacey, for sharing your experience. I just wanted to mention that and that some of these communication opportunities are available in some of the resources I talked about before, but just wanted to jump in there, share that.
Great, yes, thank you. Thank you both. A lot of great information there. We do have a question. What is the holding time for microcystin and cylinder spermopsin? Any issue with collecting finished water sample on the same day as raw samples / bloom discovery? Amanda, would you like to take that one?
I'm sure, yeah. So at least in New Hampshire with the JCLC, we're only running ELISA, and the samples can just simply be frozen. However, in this emergency situation, we would recommend maybe a lab like Green Water who could do something even better with like LC-MS and test for multiple toxins. But I don't see an issue with collecting the finished and the raw on the same day. I think that's efficient. But the microcystin, again, and cylinder spermopsin, I guess, is quite stable and can be frozen, for weeks in a sample. So there's no issue with, getting it the same day, fresh, and bringing it over. Part of the extraction process that we use for ELISA is a freeze thaw. So it's good just to grab a sample and freeze it as long as you have the appropriate jar. And in this case, what we use is the high density plastic amber bottles, but you want to be really careful if you're trying to use a different method. There are some other restrictions, like maybe wanting to use glass instead of plastic, and there's some preservatives that might be involved as well. So checking with the lab is really important.
Great, thank you, Amanda. Any other questions at this point. Okay, so there is another question, says, when are we talking about carbon? Are we adding filters on or adding carbon to the raw water and filtering it out? Question for Mike Unger.
So, oh, when we're talking about. So this would be on a temporary basis, just adding it to your existing. And John's from Canaan, and you have slow sand. I don't think that, I don't know, is Tom still on the?
Yes, Tom should
I
still
might be
be
able
here.
to speak to adding carbon to slow sand if that's a problem with clogging. As I'm picturing just in the a conventional plant, adding it at the coagulation step.
Tom?
Yeah, hey, I'm here. Yeah, for slow sand, I know Salem has quite a bit of experience with that. I think what they did was they converted, they had some roughing filters that I think they converted over to essentially like pre-sed ponds, where they added the pack. I'm not sure if Lacey's still on, but yeah, but for conventional, yeah, for sure. It's I mean, you can add it anywhere, and I guess that's one of the big, well, it's good to know what portion of toxins is extracellular versus intracellular, because if you're adding it to the raw water, but most of the toxins are intracellular, you're not going to get much benefit from adding PAC there. So I mean, I think it is kind of a case by case basis, and depends on what kind of infrastructure you have to add PAC. I think it will, it'll definitely complicate downstream treatment processes. So, I think it probably could lead to some shorter filter run times if it's not properly settled out. But yeah, I know that's not a direct answer, but I really think it's, it's probably a case by case basis.
So this
And
is Lacey.
John,
I can speak to Salem. So essentially we just have the slow sand filters, but we do have the one filter that we use as a roughing filter. We traditionally use that for turbidity events. But we did add PAC to our intake channel. And then we kind of have a long intake channel before it gets to our slow sand filters. We turned that into a sed basin. So as soon as the water came in through the intake, we put inline solar bead mixers, added PAC to try to get some contact time in that channel, put it to this south basin, which is kind of where our intake channel ends, and turn that into a sed basin, and then put the water to the roughing filter for a pre-filter. And as I mentioned before, it totally clogged our roughing filter. We had a really hard time with it. But we did initially also start using acetic acid as an additional carbon source to help fuel our smokestack. And we still continue with that, the acetic acid part. We found that to be pretty effective. But we've not been running the PAC. We've just been using other treatment options. One of the things that we did is we did boost chlorination following treatment. And then we put in a dechlorination station further downstream from our transmission mains. So we would boost chlorine up, and then right before it got to our first customer, we would bring the chlorine residual back down to a normal operating level. So I would say those are the two consistent treatment options that we've used up until we'll get the ozone online in January.
Thank you, Lacey. Thank you, everyone. That's great information. Does anybody have any more questions regarding this scenario number 2? Okay. So thank you, everybody, for that. That was great. And going back to about communication, just kind of think of it this way. Knowledge is power for people. Giving them information gives them, the public, the option to decide what they want to do, whether they want to, even though, you know, it may be safe for them to drink, they may decide not to. So again, the more information, as Lacey said and Katie said, that we can put out there in case of an event like this, the better. Okay, so moving on. So last scenario. So now the cyanotoxins are above the health advisory for all populations in finished drinking water. So on August 23rd and 24th, more finished water samples are sent to the lab for toxin testing. On August 26th, the White Mountain Water System finished water samples show microcystin concentrations at 1.9 micrograms per liter, which is now above the school-age children and an adult health advisory, which is 1.6 micrograms per liter. So now everybody, it's above the health advisory for everybody in the population. On August 29th, the citizens of White Mountain, Smithville, Jonesville, and Bear Village are preparing for days of no water. The utility resources needed include sampling, technical expertise, and labs. And then finally, at the end of all this, on the scenario on September 9th, White Mountain finished water samples are below the health advisory for all populations. So the highlight in this one is the breakthrough toxin in finished water is above drinking water health advisory levels for all populations. So now let's get on to our questions. So what emergency response actions would you take, including treatment and monitoring? And I'm just waiting for a few more votes. Okay. So just about everybody said all of the above. And we do have a couple that said issue do not drink water. Okay, our next question. What factors should be considered to determine the optimal dose of PAC to remove extracellular toxins? I'm just waiting for a couple more votes. Okay, so we have majority for both 1 and 2, and there was a couple for neither 1 or 2. Okay. The next one. What operational considerations should you consider if using slow sand filtration? Okay, so majority say both 1 and 2, and we have some for number 1 and some for number 2. The rate at which chlorine degrades microcysteines is dependent on what factors? Okay, we have some for answers 1 and 2, and some for answers 1, 2, and 3. And we have just 2 more. If you were in a situation where you had to distribute bottled water, how would you do that? Okay, so we have some for call local emergency management and some for all of the above. And the last question. At what point do you resume service to your customers? Okay, so we have some for a few. Great. Okay, so for what emergency response actions would you take, including treatment and monitoring? So first and foremost, of course, there would be issued a do not drink order to all populations. And then Mike, if you could elaborate on the rest of this, please.
Sure. So at this point, I think understanding that the raw water's clearly above the health advisory, trying to get the level of toxins down throughout the plant at each stage. We lost the
Oops.
slide there, Liz.
Sorry.
But backwashing, filters, draining the clear well, and then also looking back at the raw water. These steps would need to be, once you know you have a clean source, trying to purge the high concentrations of toxins from the treatment process. And then re-optimizing or trying to optimize treatment and essentially start from a clean slate there, stage by stage or unit process by unit process. And the same thing with flushing, the idea that if high concentrations of toxins have been out in distribution for potentially several days now, flushing, of course, won't be effective until you have a clean source to flush with, but knowing up front potentially what the residence time in the system is and key points to flush in order to make sure that you've purged that higher concentration water from the system and brought fresh water in to replace it, because the raw water and the distribution entry point might be clean or well below the health advisories, but that toxin's still out in the system from earlier.
Right. Thank you, Mike. Does anybody have any questions at this point before we go on to the next question?
Liz, I did see a question in the chat from Zeke from a couple minutes ago. He said, isn't the water still fine for toilets and maybe hand washing?
Great question. Mike, what would you suggest at that point? Or Tom?
Well, what I heard earlier this morning, that the health advisories are based on oral pathway, so contact would be okay.
Hi,
This
this
is a
is
do not
Leslie from EPA. Maybe I
Hey,
can help
Leslie.
you.
Yes. Oh, hi, Leslie.
I'm sorry, Mike, continue, but I just want to let you know that I'm here in case.
Thank you. Go for it.
Yeah, as I thought about that question, I was thinking that the person may be thinking about a do not use advice or sign instead of do not drink. So when we're talking about do not drink, it's ingestion of water. So the rest of the uses of that particular source is okay. But if you say do not use, then the rest of the uses are, prohibited as well. So when we talk about a do not drink, what we refer to is that like ingestion. So you can still use it to flush your toilet, of course. You can use it to wash your hands, except if they're kids or very toddler boys or girls, right, that they tend to put their hands in their mouth. So that could also be recommended. So small children are, not suggested to wash their hands with that particular water. But as long as it's not ingested, then the water could be used.
Great. Thank you so much, Leslie. Thank you, Mike. Okay.
Liz,
Yes.
This is Katie Foreman. Sorry, just want to jump in here too.
Oh,
just
absolutely.
build on what Leslie said.
Sure.
Just so that folks know there are places to get this information too. In the templates I mentioned earlier about the risk communication, we have templates for issuing health advisories, and they specifically go through telling consumers what is not recommended and what they can still do, as Leslie just mentioned, in using that information about the ingestion as the guiding principle there. And it has, you can use it to wash your hands, those kinds of things, the kind of questions you're going to get from the public. I just wanted to plug that so that folks know that you don't have to develop that language on your own. It is developed in our templates if you want to take a look at it.
Oh, great. Thank you so much, Katie, for letting everybody know that. That's great. Okay, for our next question, what factors should be considered to determine the optimal dose of PAC to remove extracellular toxins? Tom, do you want to elaborate on this a little bit?
Yeah, hey, yeah, so this is just kind of references the presentation I had earlier. The type of carbon, more so the pore size distribution relative to the target toxin. So for microcystins, that molecule is about 1 to 3 nanometers, and the mesopore fraction of the carbon pore distribution would be the most effective. So type of carbon that would have the most mesopores in that situation would be the best. Usually that tends to be wood, but it can be other source materials or blends. And then the amount of background organic material that can compete for absorption sites with the toxins, and then reduce the total absorption capacity of the pack. So that's the answer for that one.
Great. Thank you, Tom.
Yep.
So what operational considerations should you consider if using slow sand filtration? Tom, would you like to elaborate on that one?
Yeah, sure. And I would probably defer to Lacey in Salem's experience here, because I think I draw mostly on that. But a lower filtration rate will allow just more time for the bacteria to develop the ability to metabolize the toxins, especially if they haven't been exposed to the toxins before. So sometimes there's a little bit of a lag phase in toxin or the bacteria in the Schmutzdecker to be able to adapt to metabolizing the toxins, and so a lower filtration rate will help that. And then more frequent cleanings, obviously. I mean, that applies to slow sand filtration, but also rapid sand filters and clarifier, sludge and things like that. So just more frequent cleanings in general in a treatment plant are probably needed to deal with the cyanobacteria biomass and such. So.
Thank you, Tom. Lacey, did you want to add anything to that?
I think it's just, yeah, I agree with both of those items that, just making sure that your Schmutzdeck is healthy. If you're in algae season and you're running slow sand, making sure that you're prepped and ready to go, making sure that your filter has had ample opportunity to ripen and be as healthy as it can be, to help with the biodegradation. So yeah, I think those are probably the two biggest points.
Thank you, Lacey. So for our next question, the rate at which chlorine degrades microsystems is dependent on what factors? And Tom, if you'd like to take this one.
Yeah, sure. So the big factors are pH and temperature, especially with chlorine in microsystems. But as I mentioned in my presentation earlier, it's the similar concept with powdered activated carbon is that other constituents in the water can compete for oxidation, and so if there's or organic matter in the water that can compete and lower the efficacy of chlorine. So and then also so to account for that, a safety factor may be necessary. If you're using like the lab-based studies for reaction kinetics, like if you're using the Cyanotox spreadsheet, and I will say, I think, I'm not sure if it's come out yet, but I know AWWA was trying to incorporate TOC as a variable into their calculations. I'm not sure if anyone's aware if that spreadsheet has been modified, but so in the future, tools may be modified in order to account for some of these competing constituents. So.
Thank you, Tom. Does anybody have any questions at this point that they'd like to ask? Please feel free to jump in. Any comments? Okay. So moving on, if you were in a situation where you had to distribute bottled water, how would you do that? And I would like to ask Stephanie Nistico here at DES if she would elaborate on that.
Yeah, so all community water systems are required to have a plan in place, your emergency plan, that includes a description of providing alternate water, which may include bottled water. So we do encourage you to have a relationship with your local emergency management director to see if they can help with finding bottled water or can provide any additional resources and help with the logistics of providing alternate water or finding equipment or other resources like that. So, so that should be one of the first things you do to find bottled water. So if you don't have a relationship with your local emergency management director, we do recommend that you establish that relationship ahead of time.
Great, thank you, Stephanie. Does anybody have any questions on that?
Liz, this is Mike. I just wanted to jump in.
Sure,
I heard it this morning
absolutely.
too. I think this is a good point of all hands on deck, and that one person, even the drinking water folks in the community, can't do this alone, and to reach out and get the experts. So there's the technical side, and the operators are going to need to be doing what the operators do best with treatment and sampling. And there's a lot, Leslie talked a lot about, I'm sorry, for Salem's experience with public relations, and then this is the emergency side of public health and the emergency response. So trying to get everybody involved and spread the work, and also get the right people plugged in.
Right, absolutely. Thank you, Mike.
So this is Lacey. If I could also jump in here.
Oh, absolutely, Lacey, go ahead.
So distribution is a huge component. And even though we were in vulnerable population, we were delivering bottled water in coordination with our food share folks. So, you know, we have people who are homebound who could not make it to our distribution sites. And so we would have people call in to a specific phone number if they needed bottled water. Information would be taken down. They would be evaluated to see if they qualified. And then we had crews from other departments, stormwater, wastewater, parks, you know, administrative staff, you name it, that would deliver bottled water to these folks that qualified for it. We also had 8 water distribution sites. Because we could not use our own drinking water, we had to get water from a neighboring community. And so we had borrowed distribution trailers from other utilities. And then we ended up just making some makeshift distribution with poly tanks on the back of dump trucks. And having those at 8 different locations for people to bring their own containers to fill up water. And with that, it's about how to convey that information out to the public, where those locations are going to be, making sure there's ease of access to those locations, so that there's enough traffic control to guide people in and out. And then also talking to them about what types of containers to bring in. It's amazing what people would bring in for containers to fill up water with. So, and that was just for vulnerable population advisory. So I cannot even imagine what an all population advisory would be like. One of the things we dealt with here in Oregon, and I don't know that it would be applicable there, but we have a recycle fee on our bottles. We did have people that were coming through our sites and getting cases of bottled water and then pulling off to the side of the road, dumping them, and then going and recycling the bottles to get their 10-cent per bottle fee. So then we had to kind of scrutinize that a little bit. Anyway, if you have neighboring communities that you could get water from that are on a different water source, you know, how would you do that? Do you have contracts in place to do that? And then also what sort of equipment you have or your neighboring communities have that you can borrow to help get out water to your folks. We have a state hospital here. We have a lot of correctional facilities here that we had to truck water into daily. So a lot of logistics to definitely be worked out just on the distribution side. A lot of coordination of staff. It took a tremendous amount of staff. As I mentioned, we had staff from all sorts of groups that were working the distribution sites 24/7. initially so we could get people drinking water. So making sure, you know, you have the staff availability as well to serve your community.
Great. Thank you, Lacey. Does anybody have any further questions at this point? Okay, so to address our final poll question, at what point do you resume service to your customers? So first and foremost, I'd like to say that the EPA, their recommendations for public water systems to manage cyanotoxins in drinking water that Katie had mentioned earlier is absolutely fantastic for getting all this type of information. Per this document, they recommend that first the distribution system be monitored or should be considered to be monitored to see if any toxins are still there, even if there's a non-detect at the entry point. And I know that this has been talked about earlier in this exercise, that the distribution system monitoring could be a key component to characterize any potential threats to the public. So EPA suggests that systems continue to monitor finished water until at least 2 consecutive samples within 24 hours or 24 hours apart show concentrations of cyanotoxins basically below the health advisory levels. Even if you do get 2 finished water samples which are below the health advisory levels, it could be very important to also test the raw water to determine if the toxins are still present in the raw water, because if the toxins are still present in the raw water, that's something that definitely is great to know, to see if possibly the toxin levels in the finished water, you know, could start to rise again. Does anybody have any questions regarding that at this point?
So just going back to Zeke's question to clarify, this is to lift the advisory, not to resume service. So they're still distributing water for other uses, but these are the conditions under which you would lift the health advisory.
Great, Mike. Thank you. And at this point, Katie or Leslie or Lacey or Tom, this was our last poll question. Does anybody want to say anything further at this point?
I have nothing else to share, Liz.
Okay.
I think everything that needed to answer that question from the recommendations document is listed here. Thank you.
Great, thank you. Okay, no further questions or comments? I'm just checking the chat now. Okay, so for our last slide, so now we were at the end of our exercise. Thank you all. We just have some questions I'd like to ask. And again, please feel free to unmute yourself. So if you could let us know, what's the most important lesson you learned attending this exercise today? Did you hear something that kind of surprised you, or something that you weren't expecting to hear? Questions you might have had prior to the event that were answered today from all of our wonderful speakers? You could put your answers in the chat as well. If you don't want to unmute yourself, you can add the answer to the chat. We're just interested. Sampling protocol, okay. Yes, and also in your participant manual that I did send out prior to this exercise, a lot of the resources and links that have been talked about throughout this event with EPA, Those links are in the back of your participant manual, and also a lot of good questions you can ask yourself. There's checklists that you can go through. So those are all referenced in the back there, so you can look at those, later. And again, all the links to all the great websites for that EPA has for reference material are there as well. So we have some things in the chat. So we have one important lesson, the sampling protocol. That's great. Never jeopardize public confidence in drinking water. Absolutely. That's great as well. And okay, any other comments on that one? Okay, so how about what resources do you think you need to better respond to a cyanobacteria event now that you have, attended today's exercise? And again, feel free to unmute yourself, join in, or put an answer in the chat. I do want to say, as far as resources, we do here at DES have a cyanotoxin grant that up to $10,000 that you could, it could help you. It could pay for developing a cyanobacteria monitoring plan. It could help you buy equipment to do the monitoring that we've talked about earlier in this, to monitor, in advance for these type of events. So if you're interested in that, just shoot me an email and I can send you out the information. It's great to have, if you don't have the money to buy some equipment, and it doesn't always have to be, really expensive, high-tech equipment. It could be basically a fluorometer to check your pigments for chlorophyll A and for phycocyanin, which is the pigment that's exclusive to cyanobacteria. So you could monitor those pigments, on a weekly basis, for a pH meter to see if your pH might be increasing, as when cyanobacteria grows, an indicator is that the pH could be increasing. Secchi disc, check your turbidity, things like that. So I just wanted to put that out there, that we do have this grant available. It's a rolling grant, so there's no expiration date when you have to have your application in by. So, please feel free to email me and I can get you the information on that. And yes, this is another great comment, excuse me, from John Coffey of Canaan. Need a lab in northern New England. Yeah, very true. To have something closer, other than just Rhode Island, to have another lab like Greenwater would be great to have up here. And so our last question, do you feel this issue is important to address in your emergency response plan? After listening to the speakers, did you maybe come into the event thinking, oh, well, it hasn't happened to me yet, or it hasn't really happened to this event like what happened to Lacey out in Salem? But now after listening to everything, it could. And I believe, and Lacey, tell me if I'm wrong, I believe that you guys never had like an event like that until it actually happened. Is that correct?
That is correct. We were the first one in Oregon to have to go through the advisory, and now there's regulation here for it.
Yep.
So, we're number one. We made that happen. But yeah, no, it was not something that we ever expected to happen. And we've learned a lot from it and really ramped up our program because of it. So the more you can do ahead of time, the better, and you'll thank yourself later if you do that.
Great, thank you. Yes, so I am getting some responses in the chat that yes, it is a good idea to maybe incorporate this into a cyano emergency into your emergency response plan. So that's great. Is there any? Yep, go ahead.
Mike, I really like the risk communication tools that EPA has here
Yes.
and shared, and we're living through a worldwide public health emergency right now, and I think we've all learned how hard it is to communicate this stuff to the general public and how easy it is to get off track. And I
Right.
heard that loud and clear from Lacey's story too. So to review that information up front and have a communications plan, who's going to be
Yes.
the face to the media, and how is that going to happen, because we're beyond the point of being able to just stay in the shadows and uh, deliver a great product that people need to survive and run their businesses, and the spotlight's being turned to drinking water, for better or worse.
Right. Absolutely. Thank you, Mike. Yes, definitely. Yes, if you haven't, when you get the chance, definitely check out the EPA's risk communication toolbox. They have great information in there. And it's great to be prepared ahead of time, like Lacey had said, because when something like this happens, it's really difficult to, try to do things as on the fly, as they say. So the more you can be prepared in advance, the better, should something like this ever happen to your source water. And we do have a question to Lacey. Lacey, did they ever put a dollar figure to your response?
There is, and I don't know what the number is. I mean, I can tell you, right? I mean, just to put it into perspective, we're just completing an $80 million ozone plant that was driven solely because of this incident. I know we have 2 ELISA machines, which I think were about $30,000 or $35,000 a piece. We had to get a cryo refrigerator to freeze the samples, which was I think another $90,000. Plus the engineering that we did to get PAC up there, and all of the sampling. I want to say we easily spent 200,000 plus just in lab sampling, just for that event. So a tremendous amount of money during and even now as a response.
Wow, great, thank you. Hi, Al. Yes, I will. I will forward the presentation to everybody so you can have a copy of this PowerPoint. So any more questions or comments from anyone?
Well, this is Pierce. Yeah, I wanted to thank everybody for participating in today's event. I thought it was really great. I learned some things, and especially thanks to EPA and the speakers who came in and are helping us today. And the recordings, the presentations are all going to be recorded, so we can make them available at some point, as opposed to just the PowerPoints.
Yes. And also, again, I'd just like to say you will be getting a follow-up email after the event, and there will be an evaluation that you can fill out for us. We'd like to get your feedback. If you could fill out, I think, just a few questions. If you could just fill that out and submit it back to us, we would really appreciate it. So I would like to thank everybody. I know Pierce just did, but thank you to everybody who participated today. Thank you to our wonderful speakers who took the time to be here and offered their knowledge and expertise in what they know and what they've learned. Thank you to the participants and the attendees for taking the time to be here today. I hope that you learned something from being here, and hopefully, hopefully you never have to endure what poor Lacey had to endure, but now, hopefully after going through this event, it'll just give you some understanding of what, what planning beforehand can do and can help in a situation like this. So with that, I will say good afternoon, everybody. And if you have any questions or anything, always feel free to reach out to us. Send me an email. If you have anything further you want to know or anything, absolutely shoot me an email. So thank you, everybody. Have a great day. Stay safe. Stay well. Take care.
Bye. Thanks, Liz.
Thank you so much for everything. Thank you. Thank you very much for taking the time to be here.
Thanks, Liz.
Thank you.
Thank you, Liz. Great job.
Thank you. Thank
And
you again,
everybody.
Mike, for everything. Thanks for all your help.
Oh, my pleasure.
Thank you, guys.
Bye,
Good
everyone.
job, Liz. Thank you.
Thank you.